This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 660, recorded on September 4th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York State, Daniel Griffin.
Hello, everyone.
Hello again,
Vincent.
Daniel.
See you again.
A week later. I just looked
you
at
look good. You look like you're surviving the pandemic.
I'm back in my office. I kind of like it here. There's a lot of people around now on the floor.
You look more professional. For those people who are doing this on YouTube, Vincent, you've got the collared shirt. You're still not shaven, but you never did shave, so you've got.
Yeah. No, I'm working on having a nice look here, and you got the background, you got the books, you know what to do too. Daniel, I just looked at the New York State figures. September 7th, 8 deaths, 710 new cases, 698 cases per day over the last week. It's an increase of 8% from 2 weeks earlier. I guess things are rising.
Yeah, it's interesting. I'm going to say you can trust those numbers. The nice thing is that I was like, I feel like I'm Ronald Reagan, trust, but with verification. I'll trust the numbers as well as I have a secondary source telling me. And because I always get the feedback from Pro Health, right? Pro Health New York does, other than New York State, we do the majority of the testing in the tri-state area. So I always get the feedback, what are our percentages? And they're staying under 1% positive. But as you're saying, we're seeing a little bit of a creep. We're starting to see outbreaks. Stony Brook had a little bbit of an outbreak. We're seeing the numbers rise in the younger folks in our area, just as we're seeing them around the country. And I'm a little worried. I was talking to, and I can bring this gentleman up again later, because the reason I was talking to him was about something different. But his daughter went out to University of Ohio or Ohio State, one of those. And I said, in the old days when you went to college, you used to get a diploma. Now, what do you get when you go to college? COVID.
Yep.
So, and what's happening, right, is these kids are getting sick at the college campuses. And if you're a teenager, early 20s, and you don't feel well and you get the COVID, what do you want to do?
Get tested.
Shelter in your dorm, of course, right? So you don't spread it. No, you want to go home and have mom take care of you. Man. So now we've got all these kids, right, flying home from all these little outbreaks. And then, so we're expecting some presents to come from all these colleges, all the kids who went off to schools and are now coming home sick with the virus.
Yesterday we spoke with Christian Drosten in Germany, and he said there are very much fewer older people being infected because they've learned to stay home and keep out of trouble. That's good.
Yeah, no, you're right, actually. And there's. I'll make sure I'll get to that. But let me start off. I know we've got a lot of busy clinicians, and they always want the bullet points. So I'm going to give you the verbal bullet points. But Kiki, with her synopsis, where she does like that write up, it's actually kind of nice. So people who want their bullet point write up can go to Kiki's TWiV synopsis. But let me start off. And this is a quotation that Ben Labrat, the founder of Floating Doctors, actually suggested. And it's from Ray Bradbury's book, Something Wicked This Way Comes. A stranger shot in the street, you hardly moved to help. But if half an hour before you spent just 10 minutes with the fellow and knew a little bit about him and his family, you might just jump in front of his killer and try to stop it. So I'm going to try to get us to all start working together again here. I feel like there's way too much conflict going on here, but also, I appreciate everyone reaching out and helping us with the Floating Doctors fundraiser, because these are people down in Panama that, a lot of people don't know. And Ben thought of this when I talked about on one of my earlier episodes, how a lot of these people in Panama I know personally. And so it's really very hard to know that they're starving, that they're having issues with access to medical care. So I'll do my plug right up front. Go to parasiteswithoutborders.com and help us raise money for Floating Doctors. But also here in the U.S., let's try to get along. So I've added a new section to my weekly update, and this is called Good News. there's plenty of bad news out there. I was so tempted to start throwing more of this in, even just today as I came across stuff. And I'm not going to mention that they shut down, college, some of the college sports because a third of the elite athletes had heart inflammation. I'm just not going to mention that. We're going to good news. And so I think many of us are a bit grumpy as this drags on. Not just you, Vincent. And so I'm going to add this first part because there is some good news, but I just want to sort of couch this. It's good news relative to how bad things could be. So, one, not that many children have died in the U.S. Okay. So we just got numbers that came out for the pandemic so far in the U.S. up until the 27th of August. And although we've seen a significant increase in the number of children in the U.S. that are testing positive for SARS-CoV-2, the virus that causes COVID-19, the deaths and hospitalizations in those 18 or under remains low. And so even though we have over 6 million cases, and even though we're approaching almost 200,000 deaths, you never want to put the word only. So I'm not going to put the word only, but I'm going to say there are 101 deaths in children 19 and under. That is fortunately much smaller than the 200,000 or so that have died in the U.S. Every one of these deaths is a tragedy, but, good news relative to how tragic this could have been if this virus acted differently and targeted young people. I'll go back to what you brought up, Vincent, there is not only are we seeing less deaths per age group. So if you're at any particular age, you're less likely to die. This article by Jeffrey Harris, I'm going to refer to in a bit. But it is true that younger individuals are at lower risk of dying. Older individuals are doing a better job. I think they're aware of the risk relative to them. And I also think across the board, we're doing a much better job in the US of protecting some of our vulnerable populations, the nursing homes, limiting visitation, testing, frequent testing, actually. Next bit of good news, I'm going to say the case fatality rate, this ties in, has dropped in the US. And I just want to take a moment and reflect on why this might be the case. And I appreciate everyone. I get a lot of communication, a lot of emails, and I really appreciate that because I think that communicating during the pandemic is critical, so that I hear other people's ideas. It sort of gives me the ability to think about my ideas in that context. Not a time to silo up. It's a time for people to talk and communicate and share their thoughts and experiences. And so I was emailing this week with Jeffrey Harris. He's another MD, PhD. About 1% of us MDs also have our PhD, and he's a professor of economics at MIT at MIT. I don't know if it's affectionately called MIT, but that's what my oldest daughter calls it. And he was actually, we were emailing a little bit about a phenomenon that people probably know about from other disciplines, the whole concept of learning by doing. And he actually wrote an interesting article that specifically focused on this issue in COVID, entitled Declining COVID-19 Case Mortality, Learning by Doing. And I think that this article may resonate with a lot of people. This is not just medicine. But when you do something for a while, there's a lot of little things that you start getting right that early on you just sort of get wrong. But so, once you're developing a certain experience, so I think, non-medical people can sort of, think this makes sense there as well. But I've been mentioning a few times that one of my colleagues at the hospital developed COVID-19. And her care was quite a bit different than I think some of the care that we were delivering back in March and April. And one of the things that just, Jeffrey and I were emailing back and forth about was that with her, here we were in that like week 4, week 5, her white blood cell count started to go up. Discussed with the team, this is right about when we start seeing infections. Let's start the antibiotics now, and then let's send off those blood cultures before the first dose. Next day, the blood cultures come back positive, but she's already on antibiotics because we know what to expect. And, as we know with a lot of things in medicine, that 24 hours of getting her on appropriate therapy because we anticipated this, that makes a difference. we're getting enough sleep, right? In general, people and things are calmer. There actually are veterans with us in the trenches. So when someone is seeing their first few cases, there's those of us with experience who can kind of walk through, okay, this is what we expect, et cetera. Because it is amazing to see that for each specific age group, we're seeing less people dying. And, as we've talked about many times, not a lot of randomized control trials that we're drawing on. But somehow the care is getting better. And so this translates to medicine where outcomes are consistently better for those that see and do more in a particular area, be it surgeons, medical doctors, nurses. And so I think our physician listeners might find that there's some insight in I'm going to attribute Jeff's comments, but also I think the listeners in other fields, after you've been doing something for a while, you develop, as some people in the lab say, you develop good hands, right? The first week I'm back in the lab, I just don't expect anything to work. So I plan few experiments, and then I figure by the second week, the training wheels can come off, and I'm back in the saddle. Vaccine trials are really up and running, and more are starting all around the world and here in the U.S. And as a teaser, I'd like to give people sort of a warning. Next week, I'm going to talk a little more at a clinician patient level about the different vaccines. I know TWiV, the science has started to come more into this, but as these trials open, I think a lot of physicians and patients are going to have questions about, should I sign up? Should I be part of a trial? If I am, which trial should I set up for? And with all this warp speed stuff going on, you need volunteers in the trials, right? I mean, they want 30,000 people in each one of these trials. You got to get those 30,000 people. You got to get them into the trial in a rather quick way. And this is still in my good news. I'm going to say one of the things that we've been working on at United Health Group is something called Readiness Cohorts. I don't know if our listeners are familiar with that, but people sign up and basically say, hey, if there's a trial, I would be interested. And then what we can do is look at certain areas of the country where we think there's going to be a high enough burden of disease, right? Because you do it in New York right now, not a lot of people getting infected. And we can trigger the emails, the correspondence to go out to these people in areas that we see as sort of burgeoning hotspots. Because if you're going to do 30,000 people, you want a certain percent of those people to get exposed to the virus, and the people with the vaccines to get less disease than people without. So there's really a lot of positive things going on, a lot of coordination here between different groups to make sure that those vaccine trials get filled quickly so we can get that information, because you got to get your vaccine trial up and running. You got to get the volunteers in there. You got to get them vaccinated, and then it takes time to look at exposures. I'm going to move into transmission testing in schools. I'm going to try to be positive. Our listeners may remember that the U.S. government has embraced the idea of using rapid antigen tests to reopen the economy, and they've already committed close to $1 billion to purchase over 100 million of some of these rapid tests. And the news that came out the last few days, which I thought was really positive, it was like, what are they going to do with those tests? I think Dixon thought they were going to burn them or something. He's not as positive as I am at the moment. But the plan was the majority of these are going to go to the governors of the different states to help support school reopenings and other critical tasks. So I think, people are listening, but I want to go back a little. Like, we give Michael Mina and TWiV a lot of credit. But now I'm going to try to get hate mail from the coastal liberal elites here. And I just and I want to point out, Deborah Birx was actually pushing for rapid, limited resulting delay testing as early as April, right? And everything they do in Washington is wrong. But remember, they're the ones that actually rolled out the Abbott ID Now for screening, really let's get back to work at the White House. They weren't testing people with symptoms, right? They were just saying, everyone who walks in the door, let's not have people with infection come in here, and there was a little bit of bad press there, I guess, and I do worry that we lost some time. But I was going back to actually some of her earlier articles in early April where she was saying, listen, we could do 1 million of these tests. We've done 80,000. What's going on? Let's start embracing rapid testing. So I feel like this is a bipartisan where we all want testing without resulting delays. So the other thing I'll talk about is we've been working with a lot of the schools, and I get emails. So thank you, Vincent, for connecting people with me so we can help with testing. And I want to share a few of the different approaches we've been using to get around the bureaucracy, right? You get on the phone with the superintendent or you communicate with the superintendent, our budgets, we already have everything in place. We started planning this, it's all ready to go. And we ask, what about testing? And they say, we don't have money or we don't have plans for that. So what we've done in several areas is we just go to the teachers associations, we go to the PTA, the Parent Teachers Association, and we basically say we are willing to set up no cost to anyone access to testing. We'll give you a phone number, we'll give you a contact person. And a lot of schools, we've actually started firing this up. So don't feel like your superintendent can block your access to testing. They may not orchestrate it at a school level. They may not orchestrate it at a school district level. But parent teachers associations, teachers associations, we're always happy to reach out. And our person who's in charge of our Let's Get Back testing program and our Let's Give Back testing program, Bonnie Simmons, people feel free to contact her. Vincent, you're welcome to put her contact stuff in the show notes, bsimmons@prohealthcare.com. And we are excited to give access to testing in our area in the tri-state with our Terminator, our Testinator program. But reach out to health groups in your area. Call your doctors. Let's try to get testing really out there.
Daniel, what do you think about New York City's plan to test in the public schools selected students and faculty twice a month?
Yeah, so that's not enough. We've been reaching out, and actually several of the schools, particularly in, I'll say, lower income areas, and part of our Let's Give Back program. A lot of the kids in one of the schools that I was working on, 80% of the attendees are below the poverty line. So, we talk about the lick a stick testing in home. A lot of these kids, the home environment is not great. And so what we're trying to coordinate with them is more frequent testing at the end of the school day. And then what we'll do is we'll do it through these high throughput platforms. And if we do get one of these positives that needs to be, indeterminate, needs to be dealt with, what we'll do is when they come back, do a rapid testing to verify if that's an issue. But yeah, every 2 weeks, it's not enough. And there's different solutions that are going to work for different areas. And there's actually a lot of the New York City schools that we're trying to jump in and help them as far as testing goes. And as mentioned, it isn't always the school district that's on board. There's various political reasons why they may or may not want to endorse or affiliate or whatever it's called. And we're not trying to endorse or affiliate or anything. We're just trying to get access to testing so that we're not sending kids into an environment where they spread the virus. And I say, try to be completely positive here, but now I'm going to step back and just, people want the truth, is I was talking to some clinicians today. Sometimes the kids don't get that sick, but sometimes the parents get that sick. And we had a lot of mothers and fathers who died this spring in New York. And, now the kids are orphans, and that's a tragedy for the children. So if you don't do the right thing by our kids, and their parents get sick and die, you have not done the right thing by our kids. So we really, come on, let's make this happen. Treatment and drug updates. There was an interesting preprint out of Spain on convalescent plasma for COVID-19. And really what this was, they took a bunch of trials that were having trouble enrolling, and they put it all together. So the title was Convalescent Plasma for COVID-19, a multi-center randomized clinical trial. And what they did is they brought a number of trials together, because you had the trial with like the 81 patients, right? And then it stopped too soon. So we're trying to get more information on plasma. Maybe if you get it earlier, it's a little bit better, but the jury's still out. And not only is the jury out, but I think we have better stuff coming. People are starting to refer to these monoclonal cocktails, monoclonal antibody, either cocktails or just specific as convalescent plasma on steroids, right? So it ties into our next topic. But it's all the stuff you want without the stuff you don't want. So they're getting all those clotting factors out, and they're just giving you the antibodies that you're after. So we'll jump to steroids with that being said. And this was a study that was published in JAMA, Association between Administration of Systemic Corticosteroids and Mortality Among Critically Ill Patients with COVID-19. And here they took 7 pooled randomized studies, and they suggested, really along the lines we're seeing, a consistent approximately 30% reduction in mortality. But I want to, like, when they say this 30% reduction, we're talking about a consistent absolute 3% reduction in mortality. So you go from a 25.7 mortality down to a 22.9. So just keep this in context. This is we're chipping away at the iceberg, right? Someone kicked the side of the Titanic, and there's a little bit of a dent. So this is not, take off your mask and go to those parties.
Why do you think monoclonals are going to be better? They're going to be more potent, less of the other bad things like the clotting factors, so forth?
That's what, so two things I think is, one is, you're going to have you're not going to have the clotting factors. I don't think we want the clotting factors. There's also going to be a consistency in the level of the antibodies, because they always try to take these studies and then do these sub-analysis where you look at levels of neutralizing antibody in the convalescent plasma that was given. And it does look like there's a trend towards better outcomes if they have higher levels. But I'm also hoping that these trials are, and it looks like they will be, targeting earlier disease. So in the little thing that I mentioned about the 81 patients, those patients were getting it in the first 8 days on average. And we think this is mainly an antiviral. Some people think it has some immunomodulatory effects. I'm not sure about that. Maybe this is a Niels urine anti-idiotype network effect, but I think we're mainly thinking of this as a neutralize that virus approach.
Is there any attempt to try prophylactic trials where you would, give a healthcare or frontline worker a bolus of monoclonal and it would protect them for 3 months? That could be helpful, right?
Yeah, I don't know if you remember, but I'm sure you don't. But there was a conference at Columbia that was sort of select group was allowed to attend. It was like the VIP people. And we had just earlier that day met with Regeneron and offered specifically to do that trial. And basically said in the same conversation, we will, we're gonna create a relationship with Regeneron to use urgent care centers to recruit early stage people, but also we pitched the idea of why don't we take high risk people, maybe people in the ERs, et cetera, and you'd be amazed. Well, you wouldn't be amazed. A lot of ER docs would love to have the opportunity to participate in that kind of a trial.
Is that happening?
It's in the works, so hopefully it's going to happen.
Good.
And actually that's where I sort of got where, we're looking at these monoclonal antibodies individually, cocktails. There's also people looking at interferon therapies, antiviral specifically designed for SARS-CoV-2. So stay tuned. There's going to be a lot more coming here. Previously we just sort of grabbed what was on the shelf and hoped it worked, but now we've got people actively working on specific therapies. All right, vaccines. I'm just going to do some basics because I plan to give that physician-patient primer next week. I think there's several audiences, right, that listen to us, right? There's the physicians, there's the patients, and then there's the hardcore scientists. Sometimes I wish there was a little more hardcore science because I want to deeper discussion into, the adenoviral vectors and, the messing with the E genes, et cetera. But I think for a lot of people, they need sort of a different level, and that's what I'm going to try to do. But just to give people, get people up to speed, there are, it's a moving target, but we have lots of vaccines. 36 vaccines are in clinical trials in humans. Over 90 are in preclinical trials being tested in animals. Just to start to lay the framework for people are probably starting to hear about phase 1, phase 2, and phase 3 trials. So phase 1 are the safety trials. These are small trials, just sort of seeing are people okay? Or is there some stimulation of the immune system? Phase 2, these are the expanded trials where you move up to hundreds of people, broader age, and again, looking at safety and immune stimulation. And as our listeners probably know, 1/2 has become the paradigm for COVID-19. We just sort of jump right into the deep end. But phase 3 is the true efficacy trial. give me neutralizing antibodies, give me surrogate markers, et cetera. But what we really want to know is if I get that vaccine, am I less likely to get sick? And so those trials are firing up. And to do these trials, the FDA is involved in the U.S. and they've got to give you permission before you can go ahead and perform these trials. So we're already starting to enroll volunteers for these trials. More trials are about to start to enroll. There's going to be several different options out there and I'm going to talk more about this next time. So just to put the path in the brain here, we're going to have inactivated and attenuated whole virus vaccines, replicating and non-replicating viral vector vaccines, DNA and RNA nucleic acid vaccines, protein subunit and virus-like particle protein-based vaccines. So next time we'll go a little bit more into this, but I've been asked to do this for United Health Group, so I'm sort of double timing. I'm going to use the primer that I'm creating for the patients and clinicians there and share it. But I think we've got exciting stuff on the horizon. And the reason I think this is so important is, as I started off with, we're going to need patients and physicians willing to support this effort so we can fill these trials. Because vaccines are not something you learn by doing, right? This is something that really needs proper trials.
Daniel, you said something interesting. You said these phase 3 trials are ramping up. But just this week, the CDC said we're going to have them licensed and ready to go by November 1. How is that possible? Every vaccinologist I know says it's impossible. What do you think?
It's called magic. So, I was having a conversation with Dr. Kaplan. He's the head of the ER out at Plainview, and he's a gentleman who actually got COVID-19 back end of March, early April, probably violating his HIPAA. But sorry, Dr. Kaplan. And he got quite sick. And then, he had an antibody test 4 weeks later, nothing. Had another antibody test because he just couldn't believe it. Nothing, And this guy was, PCR positive. He was out of work for weeks. We were talking again today. He's like, Dad, I couldn't even watch TV. I was so sick. And that's what you do when you're sick. You watch TV. He's like, I couldn't even look at a screen. So he was basically saying, I want a vaccine. I don't care. I was so sick last time. I'm willing to roll the dice. And I'm not sure that's so prudent as I try to lay things out. Okay, so some vaccines have already started or already did start trials in Brazil and other parts of the world, in Europe. The approval process outside the U.S. is a little bit quicker, right? And this is the thalidomide thing that we always learn about, where sometimes something gets out there and everyone thinks it's great, and then you realize a year later, oh my gosh, what a tragedy. And the thalidomide, our listeners may not know about this, but this was a medicine that was used to treat pregnant women with nausea, with vomiting. And the impact it had is when the babies were born, a certain percent of them were born without limbs, right? The hands and feet were just basically attached at the trunk. So it interfered with development. And this was just tragic. I mean, these individuals spent their life with hands and feet attached to the trunk. That's not going to happen with vaccines. Vaccines are not going to, I don't think, cause these things. But as a lot of our listeners probably know, the virus, you always say on the catchline, viruses, the ones that make you sick. But coronavirus, it's the immune response that makes you sick. And so what we're doing with these, vaccines is we're giving you the immune response. And then we're just hoping it's not the immune response that makes you sick. And so, are you going to have antibodies that are going to cross-react with your vascular, with your blood vessels? Are you going to end up paralyzed, as we saw when people developed antibodies? Suddenly they couldn't walk and they need to be on a ventilator. We may start to get some preliminary efficacy data in the next few months, but we're not going to get the safety data. It takes time. Things take time. I know we're in the I want my information now generation. But, just like my quote from last week from one of Mao's compatriots, Zhou Enlai, it's going to take time. We will not know by November how safe these vaccines are. And so I think that's a little bit of a little frightening to a lot of people I talk to, that these vaccines are going to be out there. People are going to be encouraged to take them. And, we already have enough people who are anti-vaccines. The last thing we want to do is if you have a disaster and you expand the anti-vaccine population, it's a disaster because vaccines work when enough of the population get it. And there was a recent survey where they said, the headline was majority of Americans say they are willing to get a COVID-19 vaccine. And it was in small print, 53% are on board. That's not enough.
Yeah.
So you drop any lower, but 53%, you're already not enough. You drop any lower, you're really not enough. So, if you rush this, you can end up really being in a bad situation.
Well, when the CDC director said we're going to do it in November, for sure, he said we're going to do it. It makes me think that they've decided to release them based on the animal studies in the phase 1-2 that have already been done. And that seems like a bad idea. As you know, Gerald Ford rushed a swine flu vaccine back in 1976, and it ended up giving hundreds of people Guillain-Barré.
Yeah. And I don't know if people have ever seen Guillain-Barré, but I've seen this in a lot of individuals, and we saw it quite often in COVID-19, where an individual would start to get better, the antibodies would come up, and now they're paralyzed. And, what is, incredibly frightening. You can't move your legs. You start having trouble breathing, and it's an ascending paralysis. So it starts with, I can't move my feet. I can't move my legs. And this disease, the antibodies against spike protein, we think, trigger this. We also see cognitive impacts. We see what looks like when those antibodies come up, people have issues with cognitive function. We see vasculitis. We see the antibodies cross-reacting and destroying platelets. This is, this is very worrisome.
Yeah, I think, I'm making a point of this because I think clinicians and scientists need to speak up and object, because it seems like CDC has already decided, but this is really inappropriate, as Peter Hotez and Michael Osterholm and many others have already said.
Yeah, they've talked about, they already have a plan for who gets it first, right? And so the idea is first it's going to go to the quote unquote vulnerable populations. They're going to kill off our nursing home patients, right? And then we're going to kill off our healthcare workers because we'll get it next. I'm not excited. This is, I was frightened back when everyone was drinking bleach and swallowing, flashlights. This is, more frightening. It's more frightening because I think actually reasonable people will think it's okay because the government has okayed the vaccine.
Yeah.
So not to be partisan, but, people, really think it through. Don't just take something because you could do permanent damage. All right. Understanding the course of COVID-19. So I'm finding that for my patients, it helps that providers actually understand the clinical course. And there does seem to be this whole specific pattern of people get better. They have a sort of a lull where they feel like it's all good, and then there's this second hump when people get a lot of symptoms. I actually admitted a person this week. It really was, it hit several things. Here's a gentleman who's a security guard at the hospital, and he was relaying to me how over the winter he worked night shift. So his job was, the morgue was full, and his job was to figure out how many of the bodies he could cram into the limited freezer space, right? So what a horrible job he does for months, right? He's doing this every night, and the death toll was really high in New York to the point where we had overflow of the hospital freezers, and we had refrigerated trucks outside. This gentleman does not get COVID. He makes it all the way through. He protects himself. He wears his mask. He uses, all the right, proper preventative precautions. But what's his other job? He's a custodian at one of the schools. So they open up the school. He goes to work, and the guy he works with gives him COVID. He almost ends up intubated 3 times, sort of barely able to stay off the ventilator. He feels better. And then he has this second hump where he develops severe abdominal pain. The inflammatory markers go up again. So it helps, I think, for people. We just admitted a woman with this today, where they get better, and then they have this hump where the hair falls out, the fatigue, the trouble breathing, the muscle pains. And so I think it helps for people that we understand the clinical course, that we don't just think they're malingering. There's a few things we've actually learned that seem helpful. It would be great if we had some more studies here, but Tylenol was surprisingly helpful for some people, particularly for the prolonged fevers. We have some folks having fevers, 8-10 weeks out, but it gradually moves down. So your week moving average just keeps getting a little bit lower. Mucomyst, or the acetylcysteine, has helped quite a number of people early on, or when they get this really thick pulmonary secretions. Antihistamines sometimes help with sleep. Sleep is often an issue, but also this weird where they say, oh my gosh, I feel like I got too much sun today, this sort of histamine reaction. The non-steroidal anti-inflammatories, I feel terrible that we held them back for a lot of people, the muscle aches, all the myalgias, just taking an Aleve before bed, or maybe twice a day, taking naproxen or naproxen or ibuprofen or aspirin or indomethacin, whatever one you want to take. People really seem to be responding well. Occasionally, we do a low dose short course of steroids when people tend to have this second hump flare. Again, it would be nice to study this. So this is all sort of shared experiences. And I'll also say to be positive. There was some articles recently about the COVID tail in children, and it is much less common in children. There have been a few reports, but that's what it is. It's a handful of reports. I check in with the head of our pediatrics program here, Jay Berger, who's a TWiV listener. Fortunately, kids are also usually spared of that. And I think that's it for my clinical update.
A couple of questions, quick questions for you. Now,
Certainly.
I think the first one is from Chuck, who wanted to know how to get in touch about the Testinator program, but I think you gave us that information. We'll provide Barbara Simmons' email, right?
Bonnie Simmons. So B Simmons at it's in our show notes now.
Okay.
So reach out to her, flood her with your requests for coordination for access to testing.
Okay. A number of listeners have written in asking for your opinion on this Bradykinen hypothesis, which was published in eLife not too long ago, back in July. And they're wondering, the paper was a mechanistic model and therapeutic interventions for COVID-19 involving a RAS-mediated Bradykinen storm. What are your thoughts on that?
It's interesting, right? A friend of mine, colleague, Alex Shalshin, he's a pulmonologist, and he and I took care of a lot of COVID patients together early on. We would sort of quiz each other, like, okay, do you think it's COVID or not? Waiting to see who had the highest, percent success of figuring out if it was COVID or not. And the nice thing was, as time went on, you basically could be like, yeah, it's COVID. It's all COVID. Until he actually got sick. He got sick himself, but he's doing better. He never had a lot of hair to begin with. So it's just, he and I have a very similar, frequency with which we need to go to the barber. But he sent me the supercomputer analyzed COVID-19, the Bradykinen hypothesis. And it is very interesting because when you look at all the impacts of bradykinin, I feel like I'm going to have to give people some context here. There's a system that is tied into both inflammation and blood pressure regulation. And this involves renin, it involves the angiotensin converting enzymes. And it actually ties in with not only constriction or dilation of blood vessels, but also their permeability. So when you start looking at all the different things we observe in COVID-19, a lot of them could be explained by a direct impact of bradykinin. And there was a paper that was published actually a little while back. It took a little while to get attention here. I think it was published in July, maybe, where they actually took lavages from patients who were sick with COVID-19, and they did RNA analysis to look at gene expression, and it actually suggested that there was this bradykinin overproduction dysregulation. So I know there's a bunch of different early studies looking at this, and this stuff I think is great. One day we won't care, but we're not there yet. But the different phases, right? When they come to us in the hospital, the virus is usually on the way out. So the antivirals, things like remdesivir, even if remdesivir was super potent, the horse is out of the barn, and now we're dealing with this whole cytokine storm. Now it's being suggested that part of this storm is the bradykinin storm. So I'm fascinating for looking at, are there ways to modulate this inflammatory response, not just for COVID, but this may be, an issue for other people that have this acute respiratory distress syndrome or other inflammatory responses.
Okay, one more from Stephen Bickle, who's the medical director at the Flagler and Volusia County Health Departments. Dr. Griffin, you recommended confirmation of positive results when doing surveillance screening testing. A, is there a rough community prevalence level you use to guide this, or just any time you're doing surveillance testing?
Yeah, there is a rough prevalence, and it's what is your pre-test probability going into this? And we went back to, I'm going to go back to May. People have to listen to TWiV. We were probably in the teens back then, 617 maybe. You're going to have to Google positive predictive value in TWiV. But I talked a little bit about, you take a test that is 99% specific, right? So that doesn't mean that 1% of everyone that comes back positive that you're missing or getting. You have to take that number and look at it in the context of what percent of the people that you're testing are potentially positive. So your prevalence in your testing population. So it's not necessarily just your prevalence in your community, but it's a prevalence in that particular population. So when you get to a prevalence in the population you're testing of less than 1%, the majority of those initial positive tests, over 50%, will be not true positives. So they'll be false. So when you're at less than 1%, then you've got to do an orthogonal approach. You've got to do a confirmatory test. Once you get to up to about 5%, now you're going to say 80% of those positives are true positives. And so those are your numbers, or sort of break-offs. Less than 1, you need orthogonal testing. Greater than 5, you're going to be positive most of the time. And that sort of, I think, helps you. As far as what is that second test, it could be the same test, in all honesty, because it's an issue with the test design. It's not an issue with the person. Some people prefer a different test to be used. That's fine. I think that's more aesthetic than actual science. What we prefer to use is a rapid test that second time, because you don't want to say like, oh, you may be positive, I'll tell you in a day or 2 or 3. You want to hopefully within 8 minutes be able to say, nope, we just checked you. It's negative. This is probably the best way to do this. And I think anytime you're going to have a testing program, you need to have an approach to what do you do if a test comes out positive in potentially a population with a less than 1% prevalence.
Right. And finally, he says he's trying to calculate sensitivity and specificity for contagiousness versus sensitivity and specificity of diagnosing the infection, because my guess is that the antigen tests are very slightly less sensitive, but actually a fair amount more specific for contagiousness. Have you or your colleagues tried to estimate these values, or have any studies been published?
So that's actually, I think we talked about it last time, sort of the white paper that I wrote up for United Health Group that we shared. And actually we're trying to put this together into a model. But one of the problems with the tests that detect very low levels of RNA is you're detecting for weeks, months, in some cases, past that infectious period. So the question of contagiousness, the antigen tests are much more specific for contagiousness, particularly if you do an anterior nares. I will say saliva actually, in the studies I've seen, seems to have very similar levels of RNA to middle turbinate, so I'm okay with saliva as well. But I think the specificity is pretty similar between the two different platforms in that 99%, 98.7% range. But the ability to recognize infection versus overdiagnose people with low levels of RNA, the antigen tests they're better. They're better tests for answering certain questions.
Great. All right, that's our weekly COVID-19 update with Dr. Daniel Griffin. Thank you, Daniel.
Oh, thank you. And thank you, everyone, for tuning in.
Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's very sunny, 85, and pretty humid again.
Yeah, humidity's back. It had gone away, and now it's back. So joining us from southeastern Michigan, Kathy Spindler.
Everybody, here it's 73 Fahrenheit, 22 Celsius, and sunny.
From Austin, Texas, Rich Condit.
Hi, everybody. You're not going to believe this, but it's 75 in Austin. And it's been raining for since last evening, maybe 12 hours. Light rain now with a high this afternoon of 79. Man,
You can go
died
outside.
and gone to heaven. This is great. Yeah.
You can go for a
And
walk.
you needed rain.
Yep.
Very nice. And
All
from
good.
Western Massachusetts, Alan Dove.
Good to be here. It's 80 Fahrenheit, only 30% humidity. It's gorgeous, clear blue skies, but there's no place I'd rather be than indoors talking to y'all.
I agree. I agree with you 100%. If you like what we do here on TWiV, consider supporting us. We love your support. Those of you who support us, we love it. Microbe.tv/contribute if you're not supporting us and you'd like to. Many ways you can do that, and you'll find out there on that website.
Speaking of which, I got actually a snail mail letter from an old college friend of mine yesterday who saw us in the New York Times and said, I know that guy, and went and listened to the Fauci episode and a couple others and wrote me a letter and sent us some money.
Very nice.
Excellent.
Great.
Postal
Yeah.
system still works.
Kathy sent us a picture of the actual newspaper. Oh my gosh, they actually printed our picture in the New York Times. How about that?
Mhm.
It was very cool.
Yeah, you were on page 3. I have to point out that I was on page 1 when we did our Grammy-winning recording.
Ah,
So I
Very
am
nice.
like 2 or 3 pixels because there were 450 of us on stage. But I saw myself.
So my wife bought me a new face mask.
Ooh, I like
oh,
it.
that's
Oh, I
nice.
like
That's
that.
good.
Scientist.
So
Very
the
nice.
problem with it has been pointed out to me on Instagram. You're not supposed to believe scientists.
You're supposed
Right.
to believe
You're
science.
supposed to believe science. So
Actually, that's a really good point.
I'm gonna
Even more so now.
Yeah.
gonna remake it because I can't wear this now.
No.
Right.
But it's pretty cool. And then I'll show you the other one. So you saw the black this week in virology. So I had a white one, made.
Oh,
Good.
nice.
Nice.
Twig
Nice.
one.
Very nice.
This one is at Cafe Press, and this is funny because it has a little, paper insert. See,
Oh,
Yeah,
that's
that's
a filter.
Cool
Yeah.
filter.
filter.
Yeah, I put filters in most of my masks.
So you take it out when you wash it, right? And then
And can you replace the papers, or what are the papers? I mean,
I bought a big box of them on Amazon.
look at this; they're double.
Oh.
Maybe I'm supposed to use one at a time. Anyway, I could cut this up and put it into Virocells, right?
Yes, right. See what grows.
Probably a lot of fungus.
Yes.
Fungus among us. If you would like to chat with a virologist, you can go to the website of the American Society for Virology, asv.org. You scroll down and you will see
chat with a virologist.
Chat with a virologist. I'm doing that right now. Scroll down and it says chat with a virologist.
Yeah, we've had several people even contact the website through me, so that works too.
There it is. Chat with a virologist. So
So
check that
does
out.
this, is there like a shift schedule for virologists to operate the chat session?
It's not a chat room. No, it's you then get a list, a curated list, and
Okay.
you can try and pick somebody in your time zone and so forth.
Okay, I was just going to say, not taking the 3 a.m. shift, just so we're clear.
So last time someone wrote in and said, do coronaviruses infect insects? And I said, probably not coronas, but some other virus in the nidovirales. And I said, but Jens would know. And indeed, Jens wrote, of course there are insect-specific nidovirals. You can start with the family Mesoniviridae. It gives a link for that at Wikipedia. A family of plus-sense single-stranded RNA viruses. Yeah.
Mesoniviridae, named because their genomes are medium-sized relative to nidoviruses.
What are the hosts? I want to see what some of the hosts are here.
Mosquitoes. looks like.
Host, mosquitoes. I'm looking at the Viral Zone site. Just says mosquitoes for under Mesoniviridae. And it's got a virion morphology that looks very much like coronaviruses, which is not true of all the nidoviruses.
Oh, these were only discovered in 2011.
No wonder we didn't know.
No, that's why.
It's a long time ago. It's 9 years, man.
Geography, Vietnam, Ivory Coast.
So far.
Okay.
So what all of these viruses have in common is subgenomic RNA, positive sense RNA, blah, blah, blah. But the subgenomic RNAs are a distinguishing characteristic of the order.
Kathy, are there adenoviruses of insects?
None are coming to mind. No. There's reptile, there's fish, adenoviruses, but Jens will tell us.
Yes, that's right.
So here's the question, Jens. For every virus family or whatever the super family designation is there a member that infects some kind of insect or arthropod, right?
I can tell you that there are what we call colloquially bug poxes.
Bug pox.
Actually, they're officially entomopox viruses.
I like that. Very good.
Okay.
Those
Bug
would have
pox.
to be very small pox.
Now, last time, it was suggested, I asked Drosten to come to TWiV once a month. He said no. And then Brianne said we could just translate. So there are transcripts of his podcast, Das Coronavirus, made in German and posted on the radio site. It's a national radio station, so they post it. And a number of listeners wrote in saying, you have these, and many of them volunteered to translate them so that we could post them in English. And I said, okay, I have to ask him if it's okay with the National Public Radio there to do that. And I haven't heard back from him. But if that's okay with them, we'll do that. And in fact, someone posted, someone sent a transcript of part of one of his episodes. Man, they're dense. They say he speaks really quickly in German. There's a lot of words. Oh my gosh. So we'll see. We'll see if we can do that. That'll be useful. Okay.
Another thing I was thinking is that, you might ask him to come on just for 5 minutes, so it would be 15 minutes of his time, pre-show prep or whatever, and just give 5 minutes, the latest,
like
major
Daniel
points.
Griffin,
Maybe. Yeah. Only shorter.
Only shorter. Yes.
Yeah.
Has anybody ever been on TWiV for 5 minutes?
I don't think so.
But this would be he's already been on 2 different TWiVs, so yeah.
But it is your 15 minutes of fame to come on TWiV, right?
Right.
That's true. How else would he get the TWiV bump?
Oh, and that reminds me, someone else, a journalist in Berlin, she offered to do bullet point summaries because she said she can read the German. She can't translate. She said, I can't do that. It's too much. Although you don't have to, you just use a machine to translate it. But she said she would do bullet points. So we'll have that too. Those are different people. Some people want a bullet point. Mostly it's the UMDs out there that want bullet points. And then other people like to read. One guy wrote to me, he's an MD, he said, I go to play and I see 2 hours and I go away. I can't handle it. I need bullet points. Okay. Okie dokie. And Kiki was doing some summaries, but she stopped. So
a lot of work.
It is a lot of work. But and then TWiV in English, which is what we do here. Someone recently volunteered to do bullet points. So I said, sure, my previous bullet pointer left. Yeah. You're welcome to try, but keep it short, because Kiki was doing too long and it gets involved and it's too much work. I said, keep 1 sentence per bullet point, no more than a page. So we'll see how that works. Okay. Now for today, we have a paper, and look, I have the bloody paper printed and highlighted because I couldn't figure out how to get the PDF to highlight. I tried everything that everyone told me. I could not fix it. God damn you, Science magazine. Why the hell do you have to do that?
So you tried opening it in Adobe, making a little modification in Adobe, closing that, and then opening it into you, and it still
It
didn't.
wouldn't let me save anything. It said something about wrong integers present. Can't save the file. I downloaded it
I don't
multiple
think I had
times.
to save it. I think I just closed it. Yeah.
Closed it and it wasn't there. It didn't work. I tried opening in Word, which is no good because it makes a graphic of everything. So science, people are trying to disseminate good information. Having you protect the PDFs does not help. I do not like it. Anyone working for science, please fix it. I had to print this out and kill 5,000 trees just for this. Well, maybe
and what I'm
5.
noticing is that the things that you highlighted are not the same things as what I underlined.
I'm not surprised. I have different goals here. Anyway,
Right.
this is the first time I've printed out a paper for TWiV in 12 years. I have tried assiduously not to kill trees. I don't think I got to say, though, this brought back memories. Because I have a see the filing cabinet behind me? Sorry, those of you listening. I have 3. They're full of Xeroxes. We used to call them Xeroxes. And I can't bear to throw them away. It's the first time, but it brought back memories, like muscle memory. Like when I go sit in the tissue culture hood, right away, everything comes back. Anyway, this is published in Science, as you may have guessed. Influenza vaccine induced human bone marrow plasma cells declined within a year after vaccination. All right, so that's really what you need to know. And it's relevant to SARS-CoV-2 vaccines. We'll go into it and to talk about why, but within a year after you get that flu vaccine, bone marrow plasma cells gone. That's why in part, you need to get a vaccine every year. And this is a study that proves it. It's out of Emory University, Stanford University, and Cell Signaling Technology, which is in Massachusetts, Danvers, which is far from you, Alan, right?
Yeah, that's the eastern part of the state.
And the first author is Carl Davis, and the corresponding office, the corresponding author is Rafi Ahmed. I want to first spend about an hour on the first sentence.
Perfect.
The goal of vaccination is to generate long-lasting protection against infection. I don't think that's correct.
I would not agree.
I think the polio vaccine, which was one of the first big vaccines developed, right? You had yellow fever vaccine, you had flu vaccines, but those were kind of niche.
Smallpox vaccine.
Smallpox. But polio vaccine was designed to prevent polio, not to prevent infection. When they did the clinical trial, they counted how many kids got polio after vaccine versus placebo. They didn't see if they got infected. And in fact, IPV, that was Salk's, its first vaccine, doesn't block infection of the gut. So it doesn't block infection, it blocks disease. Now, that's not the case for all vaccines, but I don't think they should say that.
But it is the case for many.
It's the case for many, but...
And it's honestly, we've said this before, it's honestly what we ought to care about, because really, if viruses are blowing in and out and doing whatever and you never feel it and nothing happens, then who cares?
Mm-hmm. And in fact, in clinical trials, that's what they measure.
Yeah.
Right?
Yeah, they measure
whether or
outcomes.
not it prevents disease.
Well, as you will see in a bit when we talk about SARS-CoV-2, it depends on the outcomes that you're measuring in the trial.
Yes.
Some of them measure disease. Some of them also measure infection. So,
in fact, in modern clinical trials for vaccines, it is routine to measure infection, even if your main endpoints are disease-related. And this is one of the things that comes up a lot in the flu vaccine efficacy literature. Because a lot of those studies where they find, this year's vaccine is only 30% efficacious. Well, efficacious is being defined as preventing infection.
Mm-hmm.
And then you look deeper and you look at an endpoint like how many of these people were hospitalized.
That's
and you see
right.
Much more efficacious at preventing you from going to the hospital, which makes it a good vaccine, but it doesn't sound like it if you're talking about preventing infection.
Right. And Keith, to realize here is that this isn't just a vaccination issue. This is sort of the general idea of memory immune responses. Is that with your memory immune responses, you are getting a response that protects you against disease should you be infected a second time. And that second infection actually may serve to boost your immune response a little bit and help improve the third immune response if you are infected again. And of course, all of this bears on sort of the reinfection data that people have been talking about.
And by the way, this science paper, to their credit, this is a freely downloadable open access paper. So anybody who's listening, if you want to download it and have frustrations at being unable to highlight the PDF, you can.
Yeah, I think it's great that you can download it, but what's the point of not being able to highlight it? What's the point? Alan, can you answer the question?
I cannot. Okay, I do occasionally write stories for science, but I'm strictly a contractor. I can't tell you what the... Actually, I had not tried highlighting in the PDF. I'm going to try that right now.
I tried. I could not.
I was actually going to email all of you and say, do any of you have a copy that can be highlighted? And I guess you don't.
It's the first time I've experienced this, not being able to highlight the PDF, though I'm not as much of a highlighting.
Just trying to think of what the downside. I mean, if you're going to give away the PDF, then that's it, right? What are people going to do? All right, so then they stick.
But just to make it clear, you can edit it in Adobe. It's that
couldn't save it.
Oh, you couldn't even save it.
No, and there's no use for me to edit it and then close it and not save it, and then
right. Okay.
Right?
Okay.
So I could not save it.
detail.
Last time, whatever the journal was, I could save it in Acrobat, but this time it would not allow me to. I tried multiple downloads from different places, and it's all the same. Got some weird error.
I can't even use the highlighting tool.
and I'm sorry to perpetuate this.
Yeah, I can use the highlighting tool, but it only highlights the bottom, the footnote annotation, first release 13 August 2020. I can't highlight any actual text. This is on Linux. So I think this is platform independent.
That's interesting. I hadn't noticed that. Yeah, same thing, Alan.
So they go on to say that most vaccines achieve this at least in part through the generation of antibodies. I'm glad they said at least in part, right? Because there are T cells involved as well. And these antibodies are maintained by non-dividing bone marrow resident long-lived plasma cells in your bone marrow. Brianne, why do these like to live in your bone marrow? My understanding is they're formed outside the marrow, right? And then they go into the marrow, right?
So bone marrow has a lot of cytokines and other growth factors that will really help those plasma cells live for a long time.
So a guy like me who's old and has no bone marrow left or very little, what happens there? When I get vaccinated, I don't have any memory whatsoever?
No, I think you still have memory.
I know, I do have some bone marrow, right?
Yes, and bone marrow includes a lot of different immune cells. It includes plasma cells, but also new developing B cells. And so I think when you're saying I don't have bone marrow, you might be talking somewhat about those new developing cells, having fewer of those as opposed to the other parts of the bone marrow.
Someone told me once that the long bones mainly lose marrow as you age, right? And they're quite empty, actually. You can't make new ones, for sure. You can't make new memory B or T cells, actually. But ones you have may be there.
Can come back.
Anyway, they say that titers and protective immunity decline rapidly after seasonal influenza vaccination, and they wanted to know why. Is it because they don't elicit bone marrow plasma cells, or do they fail to become long-lived, et cetera? So they wanted to look at that. A very cool clinical study of healthy adults 20 to 45 years of age, and they were able to get paired bone marrow samples before and 1 month after influenza vaccination, and in some cases a third bone marrow draw a year later. This is not trivial.
No. This is some gutsy volunteers they got here who I hope were compensated for having bone marrow taken on a frequent basis like this.
And they also took blood samples, of course.
Yeah, of course.
As well. They ended up with 53 volunteers. And this was done between 2009 and 2018 with the seasonal inactivated influenza vaccine, which we should point out is not adjuvanted. And that could make a difference. So they look at immunity prior to vaccination, right? Then these people are immunized, and they use ELISpot assays to quantify the bone marrow plasma cells that are specific for influenza virus. These are secreting antibodies, both IgA and IgG, and they could easily see these in volunteers before they got the flu vaccine. And
so that's That is a memory of what?
So
running through my mind in this whole thing is original antigenic sin. Okay, and I don't know how relevant that is, but
could be vaccination, prior vaccination, or maybe not, because this study would say it's not prior vaccination.
Exactly.
Right.
Or
Or
I'm asking the question.
prior infection, I would guess.
Could be prior infection. I don't know if they sort of drilled down on things enough to discuss original antigenic sin.
No, it never. Well, we never.
It doesn't come up, but I'm not sure if they were looking specifically enough.
Interestingly,
right? No, because they don't specify what these, other than that they're against flu. They don't specify. They don't say anything specific, or do they?
They're largely against HA, specifically an H1HA.
Okay.
And so that is the antigen that they coat on the ELISA or the ELISpot plate when they're trying to look for flu-specific antibodies. So they are the H1 specific antibodies. They make a comment somewhere that in a number of different years, the H1 component was the same in the vaccine, which means they could use the same H1 to test people who had been vaccinated in different years.
And by the way, I just discovered another defect in this PDF, which is that you cannot search text.
Ooh.
You know what else is it? When I printed it out, look what happened. It's all underlined.
Oh,
see?
okay.
This is looking like a screw up. So if anybody at Science or AAAS is listening, I'm sure there must be somebody there. If you could relay a message that this particular paper, at least,
it's not the only one, but mine, when I printed it, it's not underlined.
Yours okay? Yeah, I printed this, and I actually asked Amy, and she said it's all underlined. And I said, I printed it thinking I could do better, and it was all underlined still. It's all underlined, every page. All right, fine, I could deal with the underlining. Kathy, on my second page, do I have different highlighting?
Some of them are coincident with mine.
Yeah, the results there. Yeah.
Good.
All right, so the mean percentage of IgG secreting bone marrow plasma cells producing influenza-specific antibodies was 0.8%. And they say this was actually higher, 5 to 10-fold higher than the varicella zoster virus plasma cells in the bone marrow, which is kind of interesting.
Yeah, they also do an experiment here where they look at tetanus-specific plasma cells. And I found that kind of interesting because in the abstract, they mentioned something about, there were no other changes to other B cells. And I was sort of confused. And I think they're referencing there the tetanus responses. And so they're showing it's not as though their entire antibody repertoire shrank during that time.
Yeah, that's right.
It's specific to these flu vaccine-induced cells.
Yep, that's right. Okay, so then they asked, does seasonal flu vaccination lead to the production of new influenza-specific bone marrow plasma cells, right? And so they immunized the people, and they took the bone marrow. What was it, after a month? Is that the first one?
28 days.
And they say the bone marrow plasma cells increased significantly to 1.9%. That's from 0.8%. And there's, of course, a range. That's 28 days post. So they do go up after immunization. And I'm going to skip over a lot. So if any of you want to interject things, feel free to, of course. All right. So are they maintained? Are these bone marrow plasma cells that are clearly induced 28 days after immunization, are they maintained? And so then that's 325 days after vaccination, another bone marrow. Well, they have to put a needle in your bone, right? They do in the hip typically, right?
Yep.
And I understand it hurts. I've never had one done.
I looked this up because it sounded gruesome to me, and I have heard that it hurts. It's basically done under a local anesthetic. So
because it was gruesome sounding, that's why you looked it up. Am I getting this correct?
Sure. Yeah.
Sure.
That's right.
You like.
So I wanted
to.
yeah, sure. Why not? No, I wanted to see. I think I know that a local is involved. I don't think it's a general anesthetic. They say that you feel some discomfort. It didn't sound like it was excruciating from the what I read. But
it must
it's
not be,
not.
otherwise people wouldn't do it, right?
No.
Well, there's discomfort and there's some risk to it. My wife was I think she's actually still in the registry for as a donor. Like when they need to reconstitute somebody's immune system, and it's very hard to find a match. So she's got her haplotype registered for that, and I was looking through what is this going to entail if this has to happen, and it's not. I mean, this is not a blood draw. This is something It's a big ask.
My understanding is that the local anesthetic only gets you so far.
I would think so. Yeah.
Dealing with the pain, that once you get in the bone, there's nothing that
you're not going to be jogging home from this.
But I mean, you could do this in animals, obviously, but you want to know in people, right?
You want to know in people.
And this is the kind of thing you can do. You vaccinate people and you can take bone marrow. So it's a doable experiment. It's my commendations to these volunteers. It's great.
Yes. And in animals, by the way, the bone marrow extraction is called ossobuco.
Good. Thanks, Alan.
That's the marrow, right? Yeah.
Yes, that's the dish.
I was never fond of ossobuco.
I'm not into it either.
Too metallic. Tastes like iron. Oh, I wonder why. All right. Anyway, so 325 days later, boom, percentage to baseline of influenza-specific bone marrow plasma cells. And same thing with serum antibodies, down to baseline. 1 year. Wow. It would be interesting to know the kinetics. Right? Is it 6 months, 3 months?
But now that baseline is not zero. Baseline is what?
Yeah, it's what you start.
What you came in with.
Yeah, it's like 0.8%, right?
Right.
And the question of whether that's what you started with or the immunizing one is that's what they address.
The bit about the background confuses me because the fact that everybody comes in with some background indicates that there is some durable memory of some sort.
To infection.
Something.
So how does that differ from this?
Well, this is a vaccine, right? That's the whole point. That memory is probably to a natural infection, which does a better job at making long-term bone marrow plasma cells than the vaccine. The bottom line is we've got to fix the vaccine, make it better, right? It's not good enough. And here's this vaccine has been made since the 1930s.
This is splitting hairs just a little bit, but the 0.8% is the influenza-specific antibodies, and 0.93% is the influenza-specific
cells.
Cells. So they go from 0.93 to 1.9.
Thank you. Yeah, that's right. I highlighted the wrong thing. I'm sure that's different on your paper. I highlighted the wrong paper up there.
Yeah, I think that Rich's point is really important. Because they sort of mention even at the beginning that the antibody levels peak right after vaccination, then they go down to a plateau. And that plateau is higher than at time zero, higher than before the vaccine. And so we don't totally know whether this 0.8 or 0.93 is the plateau or whether it's back down to time zero. But you'd have to find some person who had never been infected and never been vaccinated to really be able to figure that out. I know there have been some studies of cord blood that have tried to get those baseline numbers, but you can imagine the challenges.
And you can't really follow up.
Right.
You're only born
so
once.
they want to know if this contraction is due to the loss of the newly generated bone marrow plasma cells right after, or to loss of pre-existing, or both, right? So to do that, they sequence the antibody genes. They call it clonotyping, and you could look at it and exactly know what the new ones are, and it's just brilliant. It's just brilliant. And what they find is that the contraction between day 28 and 1 year is the loss of newly generated cells after vaccination, and so that's amazing. The ones that were there. The ones that were there to begin with stayed after a year.
In fact, they even talk a little bit about the loss of the newly generated BMPCs. The cells happened largely within 7 months post-vaccination. So even shorter than a year based on the serum result.
Yeah.
Well, this actually aligns pretty well with previous studies on antibody titers after vaccination and ongoing advice of you need to get a new flu shot every year, even if it's the same strains. And here you can see that the immunity really is, as far as we can tell, fading.
And this has been in 20 to 45-year-old adults, right? I bet it's even worse in us, in me and Condit. Just the two of us. We're the oldest.
Oh.
I'm just being nice, Kathy,
I qualify for the high dose vaccine, which I'm going to get on Wednesday.
I bet it goes down really quickly. Yeah. Yes, I'm going to get a high dose. And I also found that I can get the Shingrix at my local pharmacy for free. That in the high, so I'm going to do that.
Boy, that's a savings. Cause mine cost me, I think it was $175 a pop.
Wow.
2 pops.
Wow, that's a lot. Yeah, they said because I have my prescription plan with, it's the same United Healthcare, which is what Daniel Griffin works for. My vaccinations are free. Some kind of benefit. Here I am, 67. I finally get some benefits. So that's the story. Vaccination does lead to the generation of new bone marrow plasma cells, and some of them persist for a year, but most of them are lost within a year. And this is so that I didn't know how you make a bone marrow, a long-lived plasma cell. And they said the cell must reach the appropriate survival niche, compete for space there. Can you imagine? They're competing with one another. The competition is not just us humans. And the cell must undergo changes in gene expression and metabolism that promote longevity. So that somehow these influenza vaccine-induced bone marrow plasma cells fail at one or more of these steps. Yeah. And it would be nice to know which.
Right. And especially since so many other vaccines don't fail nearly so fast.
Yes.
I mean, you get other vaccinations when you're a kid and you've got immunity for decades afterwards. And then you get your flu shot and 7-8 months later, you forgot you had it.
They mentioned that the Hep B vaccine is similarly declining in the first year.
I was wondering about that because they say, first year post-vaccination. But if I'm not mistaken, the Hep B protocol is like a series of 4 vaccinations.
It's 3.
It's 3.
It was 3.
Okay. So I wonder, after 2 boosts after the initial one, whether you have durable memory.
I wonder what.
Interestingly, too, though, they say that the Tdap, tetanus, diphtheria, and acellular pertussis declines more slowly, only 40 to 66% in the first year, and that probably relates to the fact that we don't have to get those but every 10 years.
So the question is, of course, why does it decline? Yeah. And they offer 2 suggestions. One is maybe adjuvants would improve the longevity. And, the U.S. flu vaccines are not adjuvanted, although they are in Europe, or at least they're available. So it would be interesting to compare. And then they mentioned what they call antibody feedback, in which pre-existing antibodies bind to the vaccine, block B cells from recognizing. That's very cool.
So my question is this the same thing that we talk about with adenovirus vectors and pre-existing adenovirus immunity? Or is that different, Brianne?
No, I think it's different. So my understanding with adenovirus and pre-existing adenovirus vectors is that the antibodies in that case are leading to the destruction of the vaccine vector before the vaccine vector can induce a response.
Okay.
Here, the antibodies that you have, the pre-existing antibodies, are binding to naive B cells. At the same time, those naive B cells bind antigen and turning off the naive B cells so they can't grow up to become good plasma cells.
Cool. Very cool.
Okay. So targeting 2 different things then.
Yes.
Yeah.
Right.
So I asked John Udell, I said, inactivated polio vaccine, human papillomavirus vaccine, as far as I knew, they induce long-term memory B cells. So is it the antigen that makes a difference? And he said there's several differences. First, the form of the antigen. For example, the hemagglutinin in rosettes versus stably assembled nanoparticles, that could make a difference. Naive B and T cells versus hyperimmunized memory anti-flu B and T cells. Adjuvant. He said the polio thing, he thinks the RNA of poliovirus is a natural adjuvant. Alum for the human papillomavirus vaccine. Most flu vaccines don't have adjuvant, including the one in Rafi's study. He says it's very unlikely to be based on the antigenic structure of the epitopes themselves. So,
yeah, I would agree with that. I know that you have previously kind of hypothesized about viruses that have a viremic phase versus those that are only replicating in the lungs. I heard a talk on Wednesday. Obviously, this isn't about viruses. It was a talk about BCG. But they were changing the delivery route of BCG to make it IV. And they had much better memory responses and a completely different immune response. And so that really sort of screamed to me about that viremia or presence in the blood might be making a big difference, and made me want to think about it a bit more.
So
some of the, I think I've mentioned this before, some of the discussion I've heard of the human papillomavirus vaccine centers on the fact that it is a virus-like particle. And the notion that a virus-like particle may give you a more durable response.
Makes sense.
And just a pure, otherwise unstructured antigen.
So, of course, the question is, what's the relevance of this to SARS-CoV-2? And, we don't know yet, but the mRNA vaccine, so the ones in phase 3, there's two mRNA vaccines in phase 3 in the U.S. I don't know. I guess the nanoparticle is an adjuvant on its own. Is that right?
Maybe, or the RNA may be able to act as an adjuvant, just like it does with the polio case.
So,
well, and I think of the fact that you're actually synthesizing the antigen on site as stimulating a T cell as well as a B cell response. Is I don't know. Well, actually, is that who was it we were talking to earlier? Shane Crotty talked about the 3 arms of the immune system.
Yeah.
Yes.
The CD4 helper cells and the CD8 killer T cells and the antibody response. Now, the CD4 helper cells, which are going to help out on the antibody response, right? They're stimulated in response to the synthesis of the antigen intracellularly. Is that correct?
That is correct.
Okay, so the fact that the mRNA vaccines do that may help out with the I'm making this up because I don't know anything. Right? May help out with the antibody response.
It seems a reasonable theory.
Yeah. So to me, the adjuvant part actually becomes more important with what you're talking about there.
Okay.
Because you have lots of proteins that are synthesized by your cells, and in many cases, you don't want antibodies or T cells made by them. In most cases, that would be bad. And so the T cells partially know when they ought to be making a response because those antigen presenting cells have some other proteins that say this is a problem. You need to deal with this. And those are actually coming from the signaling that the adjuvant will provide.
Right.
Okay.
So we have 3 in the U.S., 3 SARS-CoV-2 vaccines in phase 3. 2 are mRNA, Moderna and Pfizer, and then the third is the Oxford AstraZeneca chimp adenovirus vector. So I don't know in terms of long term. I don't know how that's going to come out, but we'll see. We will see.
So I was trying to recall today the operation. I haven't looked this up recently. Operation Warp Speed. We've discussed this before. I think have identified 5 different sort of lead candidates, if I remember correctly, in addition to the ones you just mentioned, the Johnson & Johnson Ad26 vectored virus, and then the I'm blanking
Novavax,
which is a protein vaccine that is formulated. We discussed this before with the it's spike with the C termini all tied up together so that it looks kind of sort of like a nanoparticle.
Right.
Okay.
Yeah. Now, since we're talking about vaccines, let's move to the vaccine story. Then we'll come back to aerosols. So I'm very worried about approval of SARS-CoV-2 vaccines in the U.S. This week, there have been a spate of stories about the CDC telling states to be ready to distribute vaccines on November 1. November 1. And there are several stories here. And one of them in, what is this, WPRI.com. They quote Paul Offit, who's a pediatrician vaccinologist at UPenn, and he's a member of the FDA's Vaccine Advisory Committee. He said, being ready is reasonable. Cutting short phase 3 trials before you get the information you need isn't. Peter Hotez said he was very concerned that the FDA would use an EUA to approve a vaccine before knowing whether it works and is safe. And he says it gives the appearance of a stunt rather than an expression of public health concern. And Michael Osterholm said he was concerned about an October surprise with a vaccine being rushed through ahead of the election. The public health community wants a safe and effective vaccine as much as anybody could want it, but the data have to be clear and compelling that the vaccine is effective and that it's safe. So
let's make clear what the difference is between an EUA and licensure of a vaccine. Because this is as much for my own clarity as anything else. An emergency use approval is approval of something that has not really completed the entire authorization protocol.
Mm-hmm.
Okay, so
Right.
maybe the data, not all of the data that would ordinarily be required for licensure are in. Okay. And I presume that in a case like this, that if this were to happen at all, and it's it would be based on preliminary results, perhaps, that look strikingly positive. Okay,
And a good recent example of an EUA and also what can go wrong with an EUA is hydroxychloroquine for
Right.
treating COVID-19, which was approved under an EUA based on what was essentially anecdotal reports and a whole lot of political pressure. And so there was an EUA issued and millions of doses of this drug were prescribed to people who would not otherwise have received it. And now we've figured out that that was probably not doing anybody much good.
now in this case, Rich, the EUA, as far as I can tell, would be based on the phase 1-2 results, because the phase 3, and we'll go through the timeline of the phase 3 in a minute, there's no way they can be ready by then.
No.
well, let's discuss that. There are some. I looked up the clinical trial sites
Yes.
on all of these, and so we're
Well,
talking about.
Let's do the Moderna, okay?
Okay, before I'd like to preface this
Yes.
with something. So the reason we're talking about this is because the CDC and FDA sent these documents to various folks saying, be ready to distribute a vaccine November 1st, right? And I think Kathy pasted in links to the actual documents, which were provided on the New York Times website, and we'll also have a link to the Times article because that's as good a story to read as any about this. If you go back a couple of weeks, Trump was talking about a vaccine being ready real soon, right? People remember this?
Yeah.
If you go back, so basically what's happening now is that now some agencies that he controls are coming out and saying a vaccine could be ready real soon, right? The guy who's running Operation Warp Speed, Salawi, I think is his name, was unaware that CDC and FDA were sending out these documents.
Wow.
Think about that for a moment. The guy who's running the effort that is supposed to produce and distribute these vaccines had no idea that FDA and CDC were going to tell people to expect them November 1st. So if this was a necessary thing to say, if this was an appropriate planning step to be ready in advance of the vaccine, shouldn't he have done it?
Monsef Slaoui.
So that immediately calls this whole thing into question. And I'm reminded of an incident last year when Trump was rambling about something about a hurricane hitting Alabama. And people pointed out quite correctly that the forecast track came nowhere near Alabama for that particular hurricane, which threw him into a rage. And then the National Weather Service was forced to release an altered forecast track map that showed a possibility of the hurricane hitting Alabama. Anybody else remember that?
Yeah, too.
Oh, yeah.
Me too.
Okay.
Yes.
This, I think, is what we're dealing with. There was a statement by the president which was unsupportable by any kind of view of reality.
Mm-hmm.
People called that out, said, no, there's no way a vaccine is going to be ready by October, November. He doesn't know what he's talking about. And now agencies he controls are providing retroactive credibility for the statement that he made.
Yeah.
I'm not.
I think I'm pretty sure that's what's going on. So I just wanted to preface that so that we don't start doing something that I recently heard called competence laundering, where we give legitimacy to an idea that is really just batty.
Yeah, and the reason why this might be more serious even than the Alabama hurricane issue is that the timing of having a vaccine ready by November 1st, of course, has potential bearing on the election.
Yes, and is exactly, I'm sure, why November 1st was chosen as the date, because that would ensure that there's plenty of news coverage of this particular story.
Kathy,
So
you were
here
going
we
to say something. Go ahead.
Yeah, I want to go back to what Paul Offit said, which is being ready is reasonable. And Rich is actually the one who put in the links to the planning assumptions, the vaccination scenarios, and the action items checklist. And when you look at those are a good thing for states and hospitals and entities to be prepared about. And if they're prepared November 1st and they really didn't need to be prepared until January 1st, that's fine. They can get even more prepared and modify and so forth. So I think from the standpoint of these documents, these may have been released earlier than they might have been ordinarily.
And I think they were released with a specific date that aligned with one person's preference that was not. So I think these are documents that should be sent out, and I agree they're very rationally written. Somebody lower down in the agency thought about this and what's going to be needed. But I think the November 1st date is entirely arbitrary and has just been generated for this purpose.
Redfield, CDC heads, told Yahoo Finance that officials were preparing for what I anticipate will be reality. There'll be one or more vaccines for us in November, December. So if it's really ready in November, if all the science says it's ready, fine. But I just wanted to go through why it's probably not going to be ready.
Why we're so skeptical. Yes.
And
I just want to elaborate on these documents just a little bit. If you read through them, what it makes me realize is that distributing 100 million doses of vaccine, in particular, if you're talking about a cold chain that is as cold as minus 70 degrees centigrade, is not a trivial task. And it would be a debacle, to have all this stuff ready to go and have the states not ready to receive it and distribute it appropriately.
Yes.
Okay? So, dates and whatever aside, and my guess is that these are simply appropriate updates of existing documents.
Yes.
And I should point out that there is this sort of, I had some back and forth with Ed Niles about this because he was with BARDA, and BARDA got involved with the distribution of the 2009 influenza vaccine during a pandemic, because that was a ramped up thing where a bunch of doses were distributed. And there was a lot of collaboration between the normal flu network and BARDA, et cetera, to get that done appropriately. And it is not a trivial task. Okay.
And the flu network,
plenty of opportunities to screw that up.
Yes. And the flu network is well practiced at this exact task.
Mm-hmm.
There are distribution companies and folks involved in this who I've talked to over the years who do this amazing, largely uncredited work of actually getting the shots to the arms. And Rich is right. It's a huge, huge undertaking. We do have the benefit of this having been done every year for the past, decades with the flu shot. But in this case, when you add something like a cold chain that's got to be kept to -70, and for the folks at home who haven't worked in a lab, -20 degrees Celsius is your regular home freezer, as long as it's not a frost-free that cycles. And -70 is a laboratory freezer that you don't have sitting around. It's not a cheap item. It's probably not at your local pharmacy right now. So that vaccine, I think it's the Pfizer vaccine that they're talking about, needs -70. That is a significant issue, and that is something that I would hope states were already thinking about. A memo like this from the CDC to say, be thinking about it, is totally appropriate, and I have no qualms with that. What I bristle at is this whole November thing.
And speaking of rushing vaccines, Brianne and Rich will remember from Wednesday, I mentioned the 1976 swine flu vaccine, which was rushed and ended up causing Guillain-Barré. Just yesterday, that somebody wrote an op-ed in the Times on exactly the same thing. Ford rushed it, and look what happened to him. Trump should be careful.
Yeah, well, and I think that this is also has something to think about with making sure that the public trusts this vaccine.
Yes,
and that the public trusts vaccines in general.
If we get this wrong, it will be so much worse than not having it.
Yep.
I mean, if the first vaccine out of the gate turns out to be, 2 months down the line, turns out to be causing some horrible effect or has some unanticipated consequence or isn't as protective as we expected it to be, then the second one out of the gate is totally hosed.
Now, I'm wondering, and I think I know the answer to this, whether any vaccine has ever been administered under an emergency use approval. And I think that's probably the case for the Ebola vaccine, right? Because you can't do trials for that.
Right. Yes, I believe that was an
So this would not be...
it's not unprecedented.
Not unprecedented.
But at this scale, it's certainly unprecedented.
Sure.
This would be like issuing the annual flu vaccine to the whole country under an EUA.
Yeah. Yep.
And that's
tough. So look, Moderna's vaccine, which is in phase 3, it started, and this is from clinicaltrials.gov, started July 27th. Their goal is to recruit 30,000 participants. Okay. Then 29 days later, they get a second dose. Now, of course, they're not all recruited on July 27th. Just can't do it. I don't know how many are
recruiting.
They're still
recruiting. I don't know how many they have recruited so far. And their primary completion date estimation is October 27th, 2022. That's when it would be officially done. They're collected all the information. Okay. 2022. So I want to also point out the primary outcome measure. There are 4 of them, but here's one that I focused on. The number of participants with a first occurrence of COVID-19 starting 14 days after the second dose of the vaccine. Okay, that's from day 29 up to day 759. That's when they're supposed to observe what's the difference between vaccine and placebo in terms of getting disease. So they're not looking for preventing infection. Although a secondary outcome measure, and there are a bunch of those as well, is participants with disease and participants with SARS-CoV-2 infection. So they've added that in as a secondary measure, but it's not a primary measure. They want to prevent disease with this vaccine. But look at the numbers. So July 27th, we are now just over a month beyond that, right? So the second dose has been given, and now we're in a period. To some people.
If somebody showed up on July 27th, the day this trial began, and was screened and admitted to the trial and put into one of the groups and received the shot
that day,
then they showed up on time for their second appointment. There could be somebody in the world who's already received the second dose in this phase 3 trial. But they're still recruiting. So there are a lot of folks who haven't received dose 1.
What I do not want to happen is have, say, 500 people and then unblind the trial and say, let's look at what happened. And they're forced to do it by the administration. that's not right. This has to go for a long period before it's evaluated.
Well, and to be clear, the 2022 date is ensuring, we're really going to keep track of this thing. Even after it's been rolled out to the general public, we're going to keep following the people we vaccinated first, watch for any problems that come up in the longer term, because that isn't an ongoing concern. I don't think anybody thinks that the trial, that they wouldn't submit results before 2022.
No, that's fine.
I mean, but
to watch
I'm totally expecting results from this to come early 2021.
30,000 people is not a trivial number to recruit. And if that's what you think to have a properly powered trial, then that's the number you have to look at. Not 1,000, not 1,500. Okay, that's the point.
You can't move the goalposts as you go.
Yeah, just like with MECFS, you can't change your criteria during the study.
Midway through.
Exactly, right. So I have no idea how many people have been recruited so far. Some of the other trials also similarly involve 30,000 people. They just started a phase 3 for the chimp adenovirus, and I read they're hoping to recruit 50 people a day.
Their goal is that's another 30,000.
Right, 50 people a day. That seems conservative, especially if you have multiple sites. You should be able to do more, but 30,000 takes a while, right?
Yeah.
Yeah,
and so to get the timeline here, let's say beginning of August, people started receiving dose 1. Beginning of September, right about now, people started receiving dose 2. 14 days, if somebody gets exposed, and that's really a huge variable here, is after that second dose, you can control everything up to the second dose, and then you turn people loose into the wild, and they don't know if they got the vaccine or the control, and they've all been advised to continue acting as if they got the control. So they're still going to hopefully be avoiding infection. You're looking for some small percentage of them that would have gotten exposed and infected in both groups, and hopefully distributed across your demographic range. So you've got some old folks and some young folks and some, whiter folks and browner folks who are getting infected and all that. And then you're going to follow up and see what the course of disease is. And remember, this is a disease that's got at least a 1-month course, like if you're lucky, right? So there's a couple of weeks for symptoms to develop, and then at least a couple of weeks for symptoms to resolve. And as we heard from Ian Lipkin a while ago, if you get a more severe case, it can get pretty rugged, and you're going to be weeks recovering. And you need to follow all that in the clinical trial, because you need to know if things get worse or better as these people progress. So this is a multi-month process.
I do want to talk a little about the recruiting of 30,000 participants for the AstraZeneca trial. Supposedly they're doing recruiting at 62 sites. And I think it was Wednesday that they announced it here at the University of Michigan. And they have 3,700 people who have expressed interest in this study. Doesn't mean that they're all going to be enrolled, but yeah, there's a lot of interest in that.
Yeah. Oh, sure.
I was just thinking they ought to do the recruiting in bars.
Depends what your target is, right?
Yeah.
Yeah. Well, you want to get at least if you want a quick answer, you need people who are going to be exposing themselves to disease.
You also have to get the right age. I mean, if you want to immunize,
you really want to do it right. You got to get the right demographics, the right age, the whole thing.
Otherwise, you can't do that. Yep, 30,000.
You need to know that it's safe in all of those different populations.
Okay.
Safe and effective.
We all want this as soon as the science lets it be.
No, but
verified.
To announce it and say it's going to happen November 1, it's just not right. I just don't think that's right.
And to pick such a politicized date is that's not.
And
an accident.
It's just as ridiculous as telling people they should vote twice to test the system.
Yes,
voting twice is illegal.
Yes,
let's just make the most sensational headlines.
Yeah.
And divert
attention from
the real problems. Yeah.
All right. Anything else on vaccines? I just want to mention these aerosol studies. So you may have seen in the news, and many of you have written in about, articles purporting that aerosol transmission, that is very small particles that travel a long distance, respiratory aerosols are clearly important for transmission of SARS-CoV-2 infection. And I picked two of them because I just want to point out their deficiency. They're all deficient. One of them, they're both in press in clinical CIA, what is clinical infectious diseases. One is outbreak of COVID-19 in a nursing home associated with aerosol transmission as a result of inadequate ventilation. The other, COVID-19 patients in earlier stages exhaled millions of SARS-CoV-2 per hour. I mean, just the title of that one. They're not measuring.
SARS-CoV-2 what?
Yeah,
Yes.
it's RNA, bits of
RNA fragments.
RNA fragments. And that's the thing. So in this one study, which is out of Beijing mainly and a few other places, they have exhaled breath from confirmed COVID-19 patients. And they find by PCR that, 26% of these are positive for RNA. And they say there are millions of copies. I don't care because, first of all, it's not infectious virus, right? That's clear. But it's not even viral RNA. When you're exhaling, your cells in your upper tract are breaking from infection. They're releasing viral mRNA as well as virus particles. So we have no idea what fraction of this RNA, this millions of pieces of RNA, is actually viral. So like viral RNA. So that doesn't mean anything. So all the news reports says, aerosol transmission is happening. No, you can't prove anything from that.
There could be some small contribution of aerosol to
there might be, yeah.
But we don't have we just, I mean, this is not measles, and if it were, we would know by now. It would have a much higher R number, and the paper masks would be doing jack against it, and it would just be everywhere, and we'd all already have it.
Yeah, I mean, there may be some contribution from super spreaders for this. That's fine, but it is.
But the super spreader, when you look at the super spreader events, they are all the ones that I'm familiar with are events where it's really easy to explain as droplet transmission. Choir practice where you're spraying droplets out, and going to a bar and spraying droplets on each other as you're talking, and hanging out. Oh, by the way, they just had the first death from the Sturgis motorcycle rally where everybody was hanging out with no masks on and bragging about how the COVID-19 is a hoax. So events like that, the frat parties, all this stuff, that's where you're getting transmission. And we just, if it were aerosol, we would have, as a significant route, we would have seen that.
I just want to point it out because in the press they say, "Oh, it's definitely aerosol, and it's been hidden. It's been being hidden, blah blah blah." But these papers do not prove it. The other one, which is the nursing home, this is basically an epidemiological observation. They say the outbreak we think is caused by aerosol because nearly everybody in the nursing home was infected at the same time, and they found RNA in dust in the mesh of the air conditioners, which of course doesn't mean anything at all. So I don't think this is definitive either. And there are others like this, and very few of them ever. I think one of them that we mentioned took some breath and put it onto virocells and said, the virus grew out, but who knew how much virus was there? So I just think you should need to be very skeptical of any claims of aerosol transmission of SARS-CoV-2.
And I saw the nursing home one as a result of inadequate ventilation. I thought, wow, is this like plaintiff's attorneys hired them to write this paper? Because clearly they're angling to assign blame here to a very specific source and based on RNA data.
Well, and also that 50% of the healthcare workers on this ward. So it's really just one of 7 wards in the nursing home that they looked at. But the healthcare workers didn't always wear masks if they weren't doing patient-related activities. So, yeah.
So I guess to clear things up for anybody who's listening, and please correct me if I'm wrong on this because I can't say that I'm 100% on this. The difference in definition between droplet transmission and aerosol transmission has to do with the size of the droplet and how far it travels. And so If you're just looking at RNA, there's no way you can go back and see what the size of the droplet was. And you might be able to think maybe some of this is going on. How far did it travel? But I'm not sure that, if virus travels 1 inch farther than the definition of droplet transmission, that means it's aerosol and we all need to freak out.
There is a right. So the folks who work on this have a cutoff size. Below this, it's an aerosol. Above this, it's a droplet, and they have to pick a number, because if you didn't, then you couldn't do these studies. So they're going to draw a distinction between aerosol and droplet when in fact it's a continuum. As a droplet comes out, if it stays airborne long enough, it'll evaporate and fragment, and you may get an aerosol coming off the droplet. And so there's this whole spectrum of droplet sizes, but we're going to group them into 2 classes so that we can do this kind of study.
Exactly. And so while we can argue about where this is compared to that definition line, as Alan said, this is not measles.
No.
Which is the classic aerosol spread.
And the way I think of it is that the transmission, the spread, the infectivity is also a continuum.
Yes.
Okay.
Yep.
And people are trying to paint this as black and white, but it's not. It's gray. And it would be interesting to have enough data in a controlled enough circumstance so that you could actually put numbers on this. I'll bet you there's some sort of like exponential decay of transmissibility as you go distance away from the person who's transmitting the virus. But we don't have the data. I don't know. But my guess is that there's some function where, apparently from what we know, where the closer you are and the bigger the droplets, the more likely it's going to transmit. And then that decays.
For me, the troubling thing is a few weeks ago, WHO said it's clear the aerosol transmission is happening, and I don't see the data. Looking at these papers and others, I just don't see it. All right. Let's do some email. First one is from Andrew from Redwood City, California, where the air quality index is 52 due to historic California wildfires. Reason I'm writing is that I'm afraid that the Trump administration will pressure the FDA into approving a vaccine for emergency use before the November presidential election. He links to an article where Trump said, we'll have a vaccine before the election. He actually said it. I predict that sometime in October, Trump will announce approval of this miracle vaccine, claim it's 100% safe and effective for all ages and ethnicities. They will misrepresent the science for maximum political benefit.
And that was August 6th when he said that. And then, just now we've got the agencies he controls coming out with a document that has a date inserted that supports this.
Yeah. To combat this misinformation campaign, I hope scientists and public health experts are ready to fairly and honestly analyze the vaccine data, giving a fact-based assessment and explain the pros cons of its use at this point. In the next few weeks, could you bring more guests on the show to answer these questions? One, if you compress a vaccine development timeline, where can the timeline be safely compressed? Where can it not? What's the gray area? What's black and white? Two, when the vaccine is proposed, how should we evaluate it? What rubric should we use? Safety, immune response, durability, what populations have been tested, etc. In a nutshell, what would you need to see in the vaccine efficacy and safety for you to feel it's ready for use among priority groups like healthcare workers, elderly? When would you and your family members get it? So many journalists and policymakers watch and listen to you, and getting this out in advance of the announcement will be so helpful to the American people and our nation. Thank you. Stay safe and stay grumpy.
So I want the vaccine Tony Fauci is getting.
Yeah, an essential question here is what is the process and who is involved in the emergency use approval? And I don't know the answer to that. Okay.
Although we heard a pretty good rundown on a lot of this process from Denise Esposito a few weeks ago.
For tests.
Yes, for tests.
I was thinking about her, and I was thinking about we ought to get her back and grill her on this one.
We may want to bring her back for this.
I was thinking of Paul Offit, since he's on the Vaccine Advisory Committee.
That would be good.
Yeah.
Now, the thing is, the advice of that committee doesn't have to be taken by the FDA, right?
Doesn't have to be, but the people at the FDA who are making this decision are adults. They're not. It's not the political appointee at the top of the agency who single-handedly makes this decision. Now, he could subsequently, approve something that had been rejected, I think. But it would be a huge override of normal channels, and it would be obvious that something was not right.
So, Alan, if the head of the FDA wanted to disregard the advisory committee and just approve a vaccine on his own, could he do that?
It not just like that. There are regulations, but at this point, we have to say, to what extent are we hypothesizing them bypassing the regulations? Right?
Okay.
Because that's something that we can envision happening in the current administration. And I don't know, but if it's to the point where the scientific advisory committee says, you really need more data, and then an approval comes through under an emergency use, I'm not getting that vaccine.
So I think two things I might be looking for were how many people had that vaccine actually been tested in. Yes. Was it close to that whole 30,000 number, or was it some sort of smaller number? And was the effectiveness based on actually showing that people were prevented from getting infected, or was it that they showed, people had some antibodies, so it must be fine?
Right. So did you run your phase 3 like a phase 3, or did you run it like a phase 2?
Yeah.
Okay, Rich, can you take the next one?
Lisa writes, "Hi, TWiV team. First, thank you so much for your work and contributions to spreading knowledge and awareness. I'm very grateful for all your podcasts and share with everyone I know. Second, my institution is implementing a return to learn program that involves testing everyone, staff, faculty, students, once per month. I personally don't feel this is frequent enough to catch or prevent outbreaks. A group at my university recently put out this work, and I would love to hear your expert thoughts on it. I'm concerned that this will be used as a reason to not test more frequently. Thank you for your time." Lisa, who is at University of California at San Diego. And unfortunately, I have not looked at, she links to a bioarchive paper titled "Modeling Testing Frequencies Required for Early Detection of SARS-CoV-2 Outbreak on a University Campus." Have any of you guys looked at this?
So basically, how many infections and how often do you have to test to make sure you're picking it up? And the conclusion is widespread testing of everyone, 100% of people on campus, every month is required to detect an outbreak when there are fewer than 9 infections going on. But a high presentation of symptomatic people can compensate in part for lower levels of testing. So her original statement, once a month? No.
Yeah, once a month, everybody.
You know what New York City is proposing for their schools, their public schools? Twice a month for randomly selected faculty and students. And at huge cost because they're doing PCR, not enough. People are up in arms.
Not enough and not fast enough. I mean, if they're doing PCR, you're not going to get results fast enough, I would think, and nowhere near enough.
And when I look at this modeling, I worry because it's talking about a university community as if all university communities are the same.
Yes.
There are some that are more residential and are really sort of tightly on campus. There are some where you have lots of commuter students and you have lots more people coming and going. You might have a hospital, you might have, lots of different ways you can imagine what a university community might look like. And I have a hard time imagining that a model can successfully come up with an answer for all of those diverse university communities.
Yeah, Dartmouth, Columbia, and Springfield Technical Community College are not the same. I mean, yeah, exactly. That's a very good point.
So the problem is that this is the limitation with nucleic acid tests, right? They're slow and expensive and so forth. And that's why we have to use rapid antigen tests, which are slowly coming online.
I would also point out this is a medRxiv paper, so it has not gone through full peer review. It's a preprint, so this should be regarded as a very preliminary analysis. And the way I would read a modeling study like this is not, 1 month is enough, 1 month interval of everybody is enough, but 1 month interval testing 100% is a minimum. Because what they say really in the paper, and I'm not down with the modeling, and like Rich, I was just kind of skimming through it now, but what they're saying here is that's what it takes to be something like 90% sure that you're going to catch this thing promptly. You'd have to test everybody every month.
Right.
So
I really like the smoke detector analogy. And if
Yes.
you haven't heard
Yes.
that was last Friday. I can't remember the episode number, but to me, it's like, okay, you can have this smoke detector, but you're only going to have it in your house once a month.
yeah.
And if we detect, that your toaster, that you burned your toast, we're not going to be able to tell you till several days later. So yeah, once a month with an expensive test is not going to work.
No, and I fully understand these colleges are desperate to reopen and have something like in-person classes because they feel like they can't charge full tuition for classes over Skype. And that's unfortunate, but the virus isn't listening to your concerns.
Kathy, can you take the next one?
Sure. Anne writes, "Hey, Professor Racaniello, an update on Cornell, which you may have already heard. My daughter recently received her PhD from the school in plant biology, so she still gets campus emails. Regards. As previously communicated, last Friday evening, the Tompkins County Health Department identified a cluster of 9 COVID-19 cases among the Cornell student population related to student gatherings where masks were not worn nor physical distancing observed. This cluster a cluster is defined as 5 or more connected cases. Over the ensuing 4 days, the number of cases grew by 12, and late yesterday evening, after President Pollock's community message was sent, the president's leadership team learned that the size of this and a related cluster had grown to 39 cases. We expect the size of the clusters to grow further over the coming days as we learn the results of pending tests. With the exception of 8 non-cluster related cases, the remaining 39 positive cases from the last 6 days mainly comprise a particular cohort of students. Student athletes were 36 of those clustered cases. While they represent approximately only 0.1% of our campus population and a very small percentage of our student athletes, it points to a dangerous disregard by a group of students for the behavioral guidelines that we established to protect the public health of our community. And I'll just comment, there was an article in the New York Times.
Damn jocks.
Well, no, and about some...
I'm sorry, we were all thinking
some freshman at Cornell who went to a party and posted it on TikTok or Snapchat or somewhere, and then was publicly shamed about it and is now very remorseful. And then Vincent, you may have gotten this too because it went out to alumni of Cornell, talking about New York state rules that are making it hard for large universities in New York to have online classes. So the guidelines require significant changes in operation whenever the number of positive COVID-19 cases over a 2-week period on a college campus exceeds 100 or 5% of the campus population, including faculty, staff, and students, whichever is smaller. So as soon as the number of positive cases exceeds 100, they don't require the university to shut down, but they require that for a 2-week period, all the teaching has to move online, dining halls move to takeout meals only, and other campus activities are reduced or suspended. And this was in a letter from President Martha Kollek. The new limit is less than half of the peak infection level that we had predicted and for which we had prepared. But we are all here now, and this is our new reality, so we need to do what Cornellians have always done when faced with an enormous challenge, rise up to do everything we can to meet it.
You know, jocks, you can get really messed up and not be able to perform for the rest of your life. if you have a serious lung infection. Daniel Griffin has mentioned that before. So really not smart.
So I think there are two things to say. There was a Penn State study that was going around yesterday where they said something like 30 to 35% of their athletes who had tested positive had signs of myocarditis,
right,
inflammation of the heart. So you can get really messed up. If I defend the jocks for just a moment, we could also say that student athletes in many cases are the ones who came back on campus first to get ready for fall sports. And so seeing more cases in them could also be related to the fact that they came back on campus first.
Yes.
But did they have to go to a party without any masks?
In this particular case at Cornell, there appears to have been an egregious...
Yes.
breach.
So, give them credit for coming early, but don't take it away again for being dumb about parties.
But at the same time, let's lay a little bit of blame on the administration for thinking that they could get away with having this group of 18 to 22-year-olds on campus, and not have any problems with the pandemic. I mean,
I can't imagine you'd tell them, hey, leave your home, come where you may or may not have, especially if you're freshmen, a big social group, but then please stay in your room.
Please stay in your room. Yes, sit in your room at Cornell, but looking out at the gorges. And
I mean,
when I was in college, I never did anything that was misbehavior by any stretch of the imagination. Right?
But there was no pandemic when you were in college.
All the rules. What?
There was no pandemic when you were in college. What years were you in college, Rich? I want to make sure I'm right.
And
you're
am
right.
suspecting that he's being a little facetious here.
Yeah. 1966 to 1970.
Actually, there was a pandemic. There was
a war.
H3N2.
So wait a second. You went to Santa Cruz, right?
And I'm still alive.
And
I went to
Santa Cruz from 66 to 70, and your implication there was that there were rules?
There was a pandemic, the H3N2 influenza, 1968. So,
that went right by me.
Yeah, you don't remember anything? No.
No, the big threat then was the Vietnam War. That was what was on everybody.
Okay,
and bad vibes.
Bad vibes. Well, we didn't have any bad vibes in Santa Cruz.
Brianne, do you have time to take that quick Jake one?
I will take the quick Jake one. Jake writes, hello, Twivers. Listening to episode 659, I feel that Dr. Drosten's dismissive attitude towards saliva testing comes from the viewpoint of someone from a society without a large population of positive individuals, especially in light of the evidence that saliva testing is almost as good as the brain tickling method. They can use the more sensitive, expensive, and time-consuming PCR tests. We, on the other hand, need to use the faster and cheaper tests to try and get the virus under control. Or am I wrong?
Well, he's not wrong Daniel just sent me an article today saying saliva is just as sensitive as the brain tickling method. I like that.
I want to see the data.
I don't know. And the Yale study that came out over a month ago said the same thing. I don't know why Drosten was dismissed. I mean, he thinks that it's less sensitive. That's what he told us. So I think that's driving it. I'm not sure it has anything to do with the rate of infection in Germany and the use of PCR and so forth. But I do think he's wrong about that.
Although it would probably shape someone's perspective to be in a country that's done a good job of controlling the pandemic and that has universal health care and that has the resources to do PCR testing on people when it comes up. So, I mean, he's coming from a perspective of relative privilege in this.
Yeah, perhaps. He's worried, though. He is worried.
Yes.
You look at figure 1A and 1B in this New England Journal article that Daniel sent, and it looks convincing to me.
And the date on this is like just a few days ago. So
yeah, give it time.
All right. I will see you all later on.
Bye, Brianne.
Barker.
Take care,
Thank
Brianne.
you.
Is that Drew University BioProf Barker on the Twitter? Thanks, Brianne.
No problem. It was great to be here. Have a great weekend, everybody.
You too. Enjoy your meeting.
Try.
This is going to be more fun. Alan, you're next.
Okay, Sam writes, Dear Twivers, your recent appropriate lambasting of Brett Gruvoir, the Assistant Secretary for Health, underlines something for me. If I may speak frankly, I think you still tiptoe around the festering rot at the center of all this. You still tend to avoid turning the full fire hose of criticism directly on the one person who's responsible for appointing all these incompetent people and handicapping the CDC. It's Trump. Dr. Racaniello, on an episode a while back, you said you were done being nice, done being afraid to offend. I don't think you are. I'm afraid you're stuck in the paradigm of being unwilling to directly attack the president because that's too divisive for a general audience, as if Trump is a real president, a professional, anything, a real person who deserves a minimal level of respect. You know he's none of those things. And as you've acknowledged many times, when politics affects epidemiology, it's absolutely within your purview to call it out. You're unafraid to attack others, even Pence, but every time the conversation is approaching a place where it makes sense to criticize Trump by name for whatever he's done, it seems like you veer off and avoid direct criticism. To be fair, I think you've gotten closer and closer. Dr. Racaniello said the administration in a recent episode and Rachel Dixon might have actually gone the full mile and attacked Trump at one point, but I still think you feel you have to treat it delicately. I want you to feel free to call Trump what he is, a public health crisis. The best thing that all your listeners can do to end the pandemic is to vote for Biden on November 3rd, and that is not a political opinion. But it still feels too political to say it, doesn't it? The thing that makes this hard for me as a listener is that when content creators treat this like a normal time, as though it's proper decorum to avoid attacking the president when you speak to a general audience, it makes me feel like I'm the crazy one. It amplifies the Kafka-esque cognitive dissonance that I've felt for the last 4 years, as we've all gotten to think of the unimaginable insanity of a Trump presidency as normal. Thanks, love the podcast, kudos, etc. Sam.
What do you actually think, Sam?
Yeah, what's your opinion on this, Sam?
Yeah.
All right. Well,
Yeah.
I
think we're doing a little better on this episode because we've been pushed to it.
I get a lot of criticism that I don't read, and it hurts me personally. But your fair point. I think we're in this situation because the administration has failed at every level. Failing. Do you remember the first press conference?
It's a top.
First press conference that the president held. He said, well, there are 12 cases and within a week they'll be gone and that'll be the end of it. I mean, can you imagine any more of shirking of responsibility? So we acknowledge it. And up until today with this nonsense and getting a vaccine out before Election Day, it's disgusting. And it has to do with science and it's inextricable interaction with politics. You can't separate them. I used to think you could. You can't, right?
So vote twice.
And neither one of your votes will count and you'll be arrested.
Right.
Yeah.
Avital writes, hello, TWiV team. I'm only a graduate student studying psychology in Pittsburgh, PA, where it's 82 degrees and too humid for my taste. I moved here from Montana, hoping for some more discussion on the dose-response relationship. A few episodes ago, I believe I heard you dismiss the idea of a dose-response relationship with SARS-CoV-2. Either there's enough virus to cause an infection or there isn't. However, I recently listened to an episode of Public Health on Call where Dr. Monica Gandhi describes a direct link between viral dose and severity of sickness. Where does the truth lie? I think she's confounding sickness and infection. There is no link between dose, as far as we know, between dose and the severity of your disease. Whether you get infected or not depends on the dose. Below a certain level, and we don't know what that is, you don't have enough virus to proceed, and maybe the immune system, the innate immune system takes care of it and so forth. And above that level, it goes, and that may even vary depending on the person. But I am not aware of any evidence saying that dose is related to the severity of disease. And I know I hear Alan in my brain.
Yeah, we've talked about this and it's come up and I keep saying I need to see some clear demonstration of this. And people have written in with examples from other species, which is interesting. And we talked about the route of administration issue. So that could matter whether it's going in one way or another. But so far, people have looked, and there's just no evidence that the dose of virus that you get affects the course of the disease with this virus.
I didn't listen to the podcast. I don't like podcasts. So the summary is this doctor, an infectious disease researcher, argues that a key to beating COVID-19 is increasing the proportion of infections that cause no symptoms. The answer may be as simple as having more people wear masks. Well, that sounds fine to me.
But there's no evidence that that's going to alter the ratio of symptomatic versus asymptomatic infections.
No.
Right. All right. Rich Condit.
Tim writes, "Hi, TWiV superheroes. Accolades are not enough for all that you achieve. I and my husky look forward to each episode. Me for the information and her because of the length of the walk. Almost makes me wish I was young enough to start another career. 3 notes. First, Daniel's regular clinical information is wonderful and made me feel particularly prepared earlier in the week with the announcement of the EUA on convalescent plasma. The potential risks not disclosed in the regular press had me yelling at various podcasts for the poorly informed journalism. Your podcasts need to be required listening by mainstream media. Second, Vincent's comment on paywall issues made me attach this essay from The Economist in last week's edition. Viruses are hot stuff when The Economist writes about it. There are several articles on viruses in the issue, but the attached one has enough meat for me. I do not believe I heard you discuss the potential evolutionary significance and impacts of viruses. I still have 600 plus podcasts to go through.
You'll find that.
I wonder if a session on that idea, evolutionary significance of viruses, might be an interesting expansion of perspective. For example, I found the placenta reference and the brain neurons reference in the essay really intriguing. Not a physics unified field theory scale, but a what if kind of thing. Maybe shock impacts of pandemics have had, maybe the shock impacts of pandemics have had evolutionary impacts in the past. Third, a podcast on CBC ideas about psychologists confront impossible thing triggering a revolution in the field. Gives a link to a podcast. Discussions around replication of results, scrutinizing and verifying results and methodology prompted some questions in my mind that I wonder if you can quiet. Open science collaboration failure to validate 64% of results of 100 experiments, admitted psychology oriented, gives me pause around validation process in natural science. References to 2 sigma and 5 sigma confidence levels, particularly referencing physics experiments that were originally judged significant that were not at a different sigma level. Most importantly, the effects of data manipulation to achieve significant results and how to shield against that. Finally, does pre-registration get practiced with vaccine candidate phase 3 trials, especially with respect to analysis of the data? Love the podcast. I send out links every week. I'm not sure how many converts I have achieved. Thanks, Tim.
Your husky is going to love this recommendation. Go back and listen to earlier episodes.
All right. Most of this is in early episodes, and in fact, we talk about statistics frequently, and I will simply remind listeners that there are 3 types of lies. Lies, damn lies, and statistics.
Yes.
You know, it's interesting, these articles he's talking about. So the Economist ran a couple of articles on viruses, and like suddenly everyone has discovered how cool they are, or many people. And this was just like, wow, they had a role in the evolution of the placenta. I didn't know that. Really? What the hell do you think we've been talking about for 12 years? So it's very funny to me that these journalists look at the stories and they're going, whoa, there's something here. And that's what he is talking about. And that's okay. It's okay to hear more people learning. But as Alan said, we've talked about the evolutionary significance of viruses numerous occasions on TWiV, but we also have an entire podcast TWiV, This Week in Evolution, which is dedicated to evolution, not just of viruses, but all organisms. And you will find episodes there.
Heavily weighted toward viruses because of Vincent and
Nels, yes.
Specifically on the placenta issue, try TWiV 472, The Riddle of the Skinks.
It's a little hard to find topics based on the titles.
Right.
But I found that if you search TWiV and whatever keyword you want, you quite frequently find what you're looking for.
And a little trick that people can use, the search form on Microbe TV works okay, but if you want a little more refinement of it and you're more accustomed to the Google box, you can search for any type of a phrase and then site:/microbe.tv with no spaces. Put that in and it will search the Microbe TV site and it'll bring up podcast episodes that are about exactly this.
So a couple others are episode 25 with Luis Villarreal. The title of that one is Viral Evolution. And 92 with Marilyn Rusink, which was at an ASV meeting in Montana. So I bet its title is something about
Montana
live at ASV.
Yeah.
So, let's see, what is it? Live at ASV in Bozeman. Yeah.
And we also recently just posted TWiV 646, Virocentricity 2 with Eugene Koonin, where he talks about virus evolution.
Good. Now, Tim, this is not good for walking your dog, but you might want to watch my virology course. It's got all this stuff and more. Virology.blog/course. They're all YouTube videos.
I think his dog would love that.
Yes.
Yeah. Well, and you can even just go through and look for the ones that have to do with virus evolution for starters, but lots of other topics there in virology.
The Virocentricity 2 with Eugene Koonin is probably 2 hours long, but it'll take you 8 weeks to listen to.
Yes. Mm-hmm. There are no short episodes with Eugene, but they're good. They're really, there's a lot to chew on.
I don't know what to say about these other things he's talking about here, validation and so forth. I don't know what pre-registration.
We've touched on some of this in the past because people have written in, and again, this is pre-pandemic episodes. People wrote in about the replication crisis, and there was some news coverage of that. And this is really a lot of this applies to fields that use a lot of statistics. In fact, virtually all of it applies to fields that rely heavily on statistics because that's where you see these types of things. And there's this whole discussion around p-hacking and other things that people do to get the data to match up. In more basic molecular-oriented virology, I mean, when I was in the lab, if it wasn't 2 or 3 orders of magnitude difference, then it was no result. So we didn't really have to do statistics because it was pretty obvious whether something worked. And those types of things, there's not been any reproducibility problem at all, as far as I know, because you can then, redo those experiments. But it comes up a lot in the social sciences, and it comes up in closer to our field in medical research. So there have been a number of clinical trials and such where, looking back at the data, it seems like things weren't quite as good as they appeared at the time, and then someone subsequently discovers that it wasn't quite like we thought it was. So there are reproducibility issues, and people are talking about them. I don't have specific episode references for those.
So there's a professor at Stanford, John Ioannidis, who has made a career publishing articles saying that most of science is not reproducible, and we have actually talked about his papers before, and I think he's incorrect. I don't think most of science is irreproducible. Right.
His term "most of science" is a very narrow definition of most of science.
All right, Kathy, can you take who's next? Kenner.
Kenner writes, yes, it is. TWiV 658, your conversational format that is so attractive to your audience. We are right there on the edge of this smart and agreeable group. A little fire in the night.
Cool. I like that. Why don't you take the next one, because that was so short.
Okay. Sam writes, Dear TWiV team, I wanted to ask a contrarian question. I think that one of the silver linings to this pandemic is the increased awareness that many people have to basic hygiene like hand washing. However, you've talked several times about how fomites are not a significant route of infection. Given that, do you think that more frequent hand washing makes a noticeable difference in slowing the spread of COVID-19? Or is it another example of, quote, hygiene theater, as you say? Note that I'm not in any way advocating that we stop washing our hands. It's obviously good hygiene for a number of other pathogens. I just wonder if wash your hands to slow the spread of COVID-19 is A, sound advice that has additional collateral public health benefits, or B, a lie that has additional collateral public health benefits. Either way, I'm going to keep washing my hands. Thanks, Sam. So I'm glad you're going to keep washing your hands because I think that's important, both for COVID-19 and for the other pathogens that you mentioned. I think
it's important to understand that fomite transmission is important in a number of infectious diseases, okay? And it's not necessarily unimportant here. I mean, we're talking about sticking your hands in your face, okay? It's a little different than spraying crap on the streets, all right?
Yeah, and the easy way to distinguish hygiene theater from reasonable advice that's based on science is this something we did before the pandemic? And wash your hands is advice that everybody in public health has been telling the public for decades. I mean, they won't stop going on about the importance of washing your hands. So yeah, that's perfectly sound, and I certainly do it. Spraying hydrogen peroxide on the sides of buildings and deep cleaning the school for a day, that's not making a significant dent in anything as far as I know.
Another one that goes along with this is the don't touch your face. You've heard don't touch your face, but it's not been hammered home nearly as hard as
and it's still so hard to. I think I just scratched my eye a second ago, and
Yes, likewise. You can scrub through a YouTube and I look at myself and how many times I've touched my face. I have washed my hands. And if you, again, think about, okay, now what's different about this is we're wearing masks. Okay, think about what the mask is supposed to do. It's supposed to keep the virus from going out of you and potentially, at some level, keep virus from coming into you. So really, both sides of your mask could be contaminated with virus. So whenever you can, when you take it off, drop it in a paper bag without touching either side of it and wash your hands. So those are my pieces of advice about washing your hands.
I think that, we don't have clear evidence for fomite transmission, but I think it's likely that in some cases it's happening because the virus can maintain infectivity for a while on surfaces. So my view is that it's really not an onerous thing to say wash your hands, especially in light of putting things in your eyes and nose. So I don't think it's a lie. I just don't think we have a lot of data on that. Whereas for droplet transmission, we have epidemiological data. And of course, we know that the virus is in droplets as well.
So I'm trying to think about this. I'm trying to think about situations where we know that I just touched my nose, where we know that the fomite transmission is important. Hepatitis A, right, is a foodborne illness.
Yep.
Okay. And that epidemiologically is commonly traced to somebody involved in food preparation who is carrying the virus and does not take appropriate precautions hygiene-wise, and a bunch of people who traffic in that restaurant get sick, and you can trace it back to that restaurant epidemiologically, okay? And there it's clear that it's fomite transmission. Now, hepatitis A, I don't think that's the main way it's transmitted, I believe. I don't think that there's a lot of aerosol transmission. I have not seen a lot of evidence that says that there's that sort of epidemiological evidence for fomite transmission, for example, in a restaurant in this case. But I would imagine that, among other things, it's overwhelmed by the droplet transmission.
Yeah, exactly.
So you might not be able to see it.
But it is interesting that we haven't yet seen a super spreader event associated with takeout.
Yeah.
True.
Right. And yet, if somebody in the kitchen had hepatitis A or the measles of fomite transmission norovirus, that is totally going to spread, and it's going to be potentially on the takeout food. I mean, there are cases where somebody puked on a carpet at a wedding banquet, and 2 weeks later, somebody at another wedding banquet at the same location got norovirus because it was still in the carpet. Now, that's a case where deep cleaning the facility would have actually been a really good idea. You should have steamed that carpet. But for SARS-CoV-2, I mean, wash your hands is just advice for every virus. And we really haven't seen anything close to that kind of fomite transmission. Yeah, it's possible, but it's not the main story.
Here's a summary from WHO. Despite consistent evidence for SARS-CoV-2 contamination of surfaces and survival of infectivity, there are no specific reports which have directly demonstrated fomite transmission. People who come into contact with potentially infectious surfaces have also had contact with the infectious person, making distinction between respiratory droplet and fomites difficult to discern. However, fomite transmission is considered a likely mode of transmission given consistent findings about environmental contamination in the vicinity of infected cases and the fact that other coronaviruses and respiratory viruses can transmit this way. So despite our criticism of WHO, that's actually a reasonable.
That's pretty sound.
Actually, now that you bring that up, when you say despite our criticism of WHO, I think that, recent events, there have been missteps at all of these agencies, WHO, FDA, CDC. At the core, I still trust them. Okay. I'm wary, because of the circumstances. And I'm also cognizant that people make mistakes. Okay. But I think in the long run, I'm optimistic. We're going to survive this, and we're going to survive it with the deep state that provides all the real information intact. By the way, this was probably off, before recording, but I've started referring to, matter of fact, I think, Alan, you got me doing this, the deep state as the career individuals who really make all these machines.
The rule of law, yes.
Yeah.
There's an op-ed in the Times today by someone who says, it doesn't matter if we elect Biden, we're not going to make it to the other side. It's very negative. But, Alan, by the way, we mentioned last Wednesday, Harold Varmus had as an op-ed in the Times saying you can't trust the CDC anymore. Now, I agree that the core people working there are good, but the leadership is the issue. And WHO is a whole different story. I do think we should support WHO. I think it's crazy that we're considering not.
I think it's accurate in the case of WHO to refer to missteps. I think in the case of CDC, it's more appropriate to talk about malfeasance.
Yep.
So.
All right. One more from Alan, please.
Okay. Let's see. What are we up to? Susanna?
Yep.
Susanna. Dear TWiV hosts, guests, Jolene and Ronald, I have to shout out a loud thank you to all of you for such an incredible podcast. I love it because of the science, the format, you all sharing your expertise, humor, and all the wonderful guests. TWiV was mentioned to me by my boss and PI of 22 years in the middle of March 2020, and I became addicted. I'm excited when I see the new episode is 2.5 hours long. I'm a plant biologist at Oregon State University, plant cellular and molecular biology and genetics, and our group is mostly focusing on basic biology of pollen development these days. My PI, John, but don't ask for permission to mention his name, mentioned to me that his first biology job as an undergrad at University of Georgia, actually probably based on this information, you could narrow down who John is if you were really interested. Mentioned to me that his first biology job as an undergrad at University of Georgia was with Kathy Spindler, and it definitely influenced his passion for science. I have to mention that many SARS-CoV-1 and COVID-19 news sources I hear are read are already old news for me due to listening to TWiV. I can't thank you and your wonderful guests enough. I think you mean SARS-CoV-2 probably. Today during my field work, sunny blue sky, about 95 Fahrenheit, 35 C, I was listening to TWiV 659 episode with Christian Drosten while trying to find out major differences between the US and German responses to SARS-CoV-2 epidemic with such different outcomes, at least for now. I could not stop wondering about the role of general population health in the two countries. Although I've heard about many cases of healthy people coming down with COVID-19, I tend to think that an overall healthier population would have an advantage in fighting the virus off in earlier stages. Any thoughts?
So is she suggesting that we have more comorbidities here in the U.S.?
That's the idea.
Yeah.
I don't know that that's true.
I mean, is the German population in general healthier than? I have no idea.
Sure, yes.
Yes.
I mean, we have among the highest obesity rates in the world.
Mm-hmm.
So I'm going to guess that you could make an argument for a healthier overall population in Germany. There might
not be as many health disparities in the range of their population as there are in ours.
Because of universal health care and because of a more tight-knit population. I think things like that could make a big difference, or maybe it could make a difference in outcomes, in number of deaths. Certainly in countries where you have an older population, you'd expect more people dying of COVID-19. If you have a younger population, you might expect fewer. But neither of those things is going to affect incidence or spread of the virus. What's making a huge difference in places like Germany is proper management. Mm-hmm. Where early on they recognized this as the serious threat that it was. They followed the science. They did the testing. They quarantined people who were symptomatic, and then they quarantined people who were asymptomatic when they got their testing ramped up. And they sheltered in place, did the whole thing right and successfully flattened the curve. And that's how Germany got to where it is with the pandemic versus the utter disaster of the U.S. response, which has been completely incoherent from the top level down. And the top level, I would be referring to Trump in answer of the earlier email, and has been left up to the 50 states to figure out how to muddle through with less significant resources than the federal government. And so here we are.
I also think that's interesting. Christian mentioned that their death rate has decreased because older people are being infected less because they're staying away. They're staying inside mostly, and they're doing it, which is what you should do, right?
They're following the science.
Yep.
So, like Rich, I've had a number of people contact me because of TWiV, and I have owed John an email since the beginning of the pandemic when he emailed me and mentioned that he'd been listening to TWiV. So hi to John. Sorry, I haven't written yet. I promise to write soon. I think John was my second undergraduate doing research in my lab. Wow. I have my timeline in my office at work, so I can't double check that. But I do remember when John and his wife visited me in Athens, and they told me about GFP. So I remember specifically we were on my screen porch and they were telling me about this green fluorescent protein and how it was going to change things. And so obviously that was well before the Nobel Prize for it and so forth. But it's funny how you remember some things that you learn in science and from whom and where, and that's one of them.
I love hearing how people just encounter TWiV and then they love it. It's like, because for many people, it's very appealing, right? And we've always known that, but unless you encounter it, you're not going to know that. So it's the initial encounter that's important, and more people now are doing it. So that's great. I do think that if more people encountered it, they would like it as well, despite it being long and deep and so forth. It's an interesting way to learn.
Need a dog.
You don't have to have a dog. You can just stop and start.
and or listen at 2x speed while you're doing something, or Yeah,
there are many ways. All right, TWiV 660. Microbe.tv/TWiV is where you'll find the text of all the letters, their show notes, links to stories, and so forth. If you want to send us a question or comment, TWiV@microbe.tv. And if you'd like to support us financially, you can go to microbe.tv/contribute, where we have Patreon accounts, PayPal accounts, and you can there's an affiliate link for Amazon shopping where we get a fraction of your purchases. There's a cafepress.com/TWiV where you can buy stuff to wear and drink from. All of that helps us.
These are not the same item, by the way.
We'd appreciate.
I don't think we have those hats with the straws coming down. Do we?
No, we don't. I don't want to do that.
Yet.
Today, someone asked me for buttons and stickers, which is easy enough.
I've had stickers in the past for the TWiV 500 because I have some stuck up in my office.
Yes, I'm going to put some of the more recent designs on stickers and buttons, like the miasma and the masks are cool, right? That's easy enough to do. I will do that for you. Okay, Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. On Twitter, he is Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.